Welcome to Recombination Nation. Today I have found another inspiring microbiologist who is working at the cutting edge of this Wild West topic that humanity has only recently stumbled into and is still blinking and looking around and trying to figure out what the hell is going on, not just in the world around us, but inside our own bodies. So today we're talking to Stephen Skolnick. Um, I've only found his work recently, and it has been utterly eye-opening. I had hints of what was going on this front, and he has taken things so much further. So I'll throw over to you, Stephen, and you can tell us a bit more about your kind of path through life leading to this point, and maybe summarize like what you think are the most interesting possible health conditions that you've linked to a microbiome origin.
Excellent. Yeah. Well, thank you so much for having me on. First off, this is the first time I've been invited to do a podcast, so really appreciate the chance to talk to you and your audience about it. Yeah. So I describe myself as a poop scientist, which usually at parties people go, "What?" And you heard me. And it's I was not always a poop scientist. I my undergrad was in physics. I went to Indiana University for medical physics, and but around the time I was graduating, it was like 2014, which was around the time gene sequencing was getting cheap enough that you could just shit into Illumina for kicks and, see what's going on down there. And that was when we recognized, like, oh, there's this whole other facet of human biology that we've basically been ignoring because what really like made that, uh, uh, I guess it was not fully understood before that the bacteria that you find in a human gut, many of them are ubiquitous, meaning they are found in practically every healthy human gut, and unique. They are not really found like anywhere else on Earth besides mammalian guts. And, I think before that, it was largely assumed that, like, okay, a lot of these bacteria just sort of come from the environment or from the food that we eat, fermentates and things like that. But no, it's very probable that, these things are passed down vertically across generations reliably, and it's not fully clear how that happens, but essentially that these things are as much a part of the human body as the mitochondrion, right? Which is, outside the 23 chromosomes, but still very much a part of the human organism. You can't really have a happy, healthy human or any multicellular life form without it. And that we sort of rely on these bacterial genes, and the way that you can sort of prove this out is looking at Neanderthal coproliths, right? Like early proto-human stuff. You find a lot of the same bacteria in those, and just from like gene sequencing, because DNA is quite stable. You find a lot of the same bacteria in those samples that you find in the modern human gut, right? And so probably these things have been with us since we've been mammals. Um, and yeah, I found all this out in an evolutionary biology class halfway through my senior year in college, and then it was like, fuck, picked the wrong major. But it's good. Graduated with a physics degree, anyways, and sort of developed my passion for this in my off time. I took a job as a science writer at the American Physical Society, or APS Physics. And that's a journal publishing society. They put out physics journals, Physical Review A through E and X. And my job there was basically just like trying to get people excited about science. I ran their public outreach website, physicscentral.com, which was a lot of fun and, great practice because I would get a summary in my inbox every day of all the new papers coming out in these physics journals. And it was like, all right, how do we, like get to comb through, find the interesting ones, read the full papers, maybe interview the authors, and really try and find a way to make these things interesting and accessible to the public without lying, right? And that's the hard part, because I can't count the number of times I've seen a headline that's like, "Scientists invent real working lightsaber," and then you click through and you read the article, and it's like the article is about photonic coupling in rubidium gas, and it's like that's not true at all. Uh, Disappointed. I wanted a lightsaber. But
hmm
we well so I but like in my so and this was a great job for me because I got to hone these sort of science communication skills. Uh and in my off time I was just like reading everything I could about the microbiome, right? I discovered Sci-Hub and was just pirating papers left and right. And sort of exploring these pathways. And a little bit of background about me. Both my parents were neuroscientists, right? They worked in pharma, they worked at NIH. My dad did some of the early work on ketamine and depression. My mom did some of the early work on like oxytocin. And mm, so I had a little bit of background in this, and we've been talking about the serotonin system at the dinner table since I was like 5, and but whenever I would ask, like, okay, where does this chemical imbalance that leads to like depression, for instance, come from? And it was sort of a shrug. It was like, well, that's above my pay grade.
Hmm.
And so when I learned of like, oh, okay, here's this whole other component of human biology that is, I would say, like, it's I think 1,000 times as many genes as you find in the human genome,
Hmm.
and half variable from person to person, right? We're all 99.5 to 9% identical to one another genetically. But the microbiome, it's up to 50% variable from
Do,
person to
do
person.
you know how I've started to look at it? That the
Hmm.
human is like the tortilla, and the microbiome is like all the different fillings that
The
you can
filling.
put into the arrangement.
I love that. Yes, yeah. And some are with beans and some are without, right?
Hmm.
Um, yeah, no, that's a good way to put it. We're all burritos, but there's a lot of variability there.
Hmm.
And so if you want to understand why one person is sick and another person is healthy, especially when we're talking about chronic diseases that seem to sort of it's like a switch that flips late in life, or even, in adolescence or something like that. Like this really seems like the logical place to look, especially if we've already checked the genome and have not like found anything conclusive there,
Hmm.
right? Mm. So, uh, you know, read a lot about it, developed a sort of broad knowledge base, eventually to the point where I started having my own hypotheses around like, oh, okay, well, here's this small molecule that's linked to biopterin recycling, and biopterin's a cofactor for some very important enzymes in your body. And so I took out a use patent around this compound, right? Because it's a natural molecule, and so you can't patent the molecule itself, but you can patent a use for it. And put a patent out on use of quine, to treat neuropsychiatric issues. And you know, at the time, my I was dating a girl who I've known for quite a long time. She had just moved up to Boston from we were in the DC area at the time. She had just moved up to Boston to do a 1 year master's at Harvard. And we're on the phone one day, and she's like, Stephen, you got to risk it for the biscuit. If you're like you're really passionate about this stuff, like everybody and their brother is moving to Boston and starting a microbiome science company. So why not do the same? So I ended up finding a lab at MIT that was doing some very interesting work on probiotics and oxytocin. And you know, uh, APS happened to be having its March meeting in Boston that year, so I booked some time with this professor and said, "Hey, can I come do some work in your lab? I'll be an unpaid intern for however." So eventually, yeah, I learned to pipette, got the basics of micro or like, really just the basics of wet lab work. Killed a lot of mice, autopsied a lot of mice during that time, which I think is just like a rite of passage in bioscience work.
Hmm.
But and that was supposed to transition to like a paid situation. Never did because of some boondoggle with the grant funding. But
Hmm.
while I was up there, I gave a talk at a conference and got connected with this guy Phil Strandwitz, who ran a microbiome science company called Holobiome that was investigating the gut-brain axis. And so they hired me on. I assigned the patent to them so that they would take care of the fees and things like that. And that was where I learned, the nuts and bolts of anaerobic microbiology, how to actually like grow the bacteria that you find in a healthy human gut. And that was, gosh, 5, 6 years ago now. Anyways, we were about to close the Series A when I joined. 3 years later, we were still about to close the Series A. And so well, I got poached out basically. I had started my blog in the meantime. Got poached out to a company called Seed that makes a probiotic. It's very popular. That's really all I can say about it because I
Hmm.
signed a non-disparagement clause in my severance contract. And they offered like double my salary, so I went and worked for them for a little while. Um, and that was where I got to actually have my own lab. Basically, they sent me out to the Swiss Institute for Allergy und Asthmaforschung in Davos, of all places. where I set up an anaerobic bacteria lab in the basement and started doing strain isolation, finding some, sort of exploring the dark matter of the microbiome, trying to get some of these things that nobody had grown in culture before yet, and had pretty good luck. Got a good number of new genera, new species. And a lot of those are still sitting in a freezer in the basement in Switzerland. But from there, once we got back to the US, really like tried to get up the momentum to develop some of these things in-house at seed. It just wasn't happening. And so I quit there, started my own company. We are Constellation Bio. That was about 8 months ago, 9 months ago now.
Hmm.
And yeah, we're here we are. We just moved the lab from Indianapolis to Berkeley a month or two ago, and we're getting things set up to go into human trials with a cholesterol reducing bacterium. So
Well, that's probably one of the broadest conditions. Did you want to briefly talk about the proposed mechanism with
yes, yeah,
cholesterol
would
metabolism
love
there?
to. Would love to. So this is one of these functions that is core to the mammalian microbiome. In that, like, you look at human stool, and you find in 4 out of 5 people there's a chemical called caprostanol in the poop, right? And this is a saturated, a hydrogenated derivative of cholesterol. I said it's a cholesterol reducing bacterium, and that's a little bit of a double entendre in that, like, it is a cholesterol reducing bacterium in the chemical sense of the word reduced, where it is a sort of hydrogenation reaction.
Yeah,
But
the
it looks
opposite
like it
of
is
oxidizing.
exactly, yeah. There's
Yeah.
oxidation, there's reduction. So it is a cholesterol reducing bacterium, but it looks like this is also a cholesterol reducing bacterium in that it lowers serum cholesterol levels. Because, well, like I said, 4 out of 5 people you find caprostanol in the stool, and this is the saturated derivative. And caprostanol, like it's a small change, but it changes the properties such that it's not reabsorbed in the small intestine,
Hmm.
right? And the human liver churns out about a gram of cholesterol every day, right? And this is put out into the gallbladder and it's put out into the intestines in bile. And it helps emulsify dietary fats. It helps sort of disperse them and helps to absorb them across the small intestinal wall. And this is part of a large part of why the liver produces cholesterol.
Hmm.
Uh, uh, the thing is that, like, there's really only two ways out of your body, right? Number one and number two. And number one only works for things that are water soluble, which cholesterol very much is not. And
Hmm.
number two starts with bile, and that's excreted at the top of the small intestine. So it's got to wind its way through like 30 feet of small intestine before it can get to the large intestine, the colon, and then leave the body. And because, cholesterol is reabsorbed very efficiently in the small intestine, if you don't have these bacteria in your gut, taking the cholesterol, transforming it into caprostanol, and sort of crystallizing it out in the GI tract, then there's no real way out of your body for cholesterol aside from
Hmm.
as like bile acids and things, which is a relatively small component. And so if you look in, the guts of horses, dogs, cats, squirrels, any wild type mammal, you will find mostly caprostanol rather than cholesterol. And so when something is present in 4 out of 5 humans and every wild type mammal that we've checked, that makes me think it's supposed to be present in 5 out of 5 humans.
Yeah.
And we've just fucked it up with antibiotics along the way somewhere. And they actually ran a study in, I think, 1990, where they pretty much proved this. They tracked levels of caprostanol in people's poop as they went on a standard, like, 6-day course of antibiotics. And they used like 10 different kinds of antibiotic. And in about in 90% of the people in their study, levels of caprostanol in the poop would drop while they're on the antibiotics, right? And as like the microbiome recovers over the course of the next month, they would, the levels of caprostanol go back up. And uh, so it's funny because their finding at their headline finding in the abstract was no significant difference by 1 month later from baseline. But
Hmm.
the thing about, I guess, statistics is that it averages out the effects, right? And in 10% of their subjects, uh, levels of caprostanol drop to zero while they're on that antibiotic, and they stay there even a month later,
Yep.
suggesting a kind of extinction event.
Yeah,
right?
yeah.
That the caprosenol producers in their microbiome have either gone fully extinct or are so suppressed and outcompeted by other things as to make no difference. And so this suggests that like the 20% of people who are missing caprosenol in their poop, yeah, maybe just got unlucky with the course of antibiotics at some point and just need that species or whatever species produce caprosenol sort of put back. Um, And so this has been known about for a good long time. Caprosenol has been observed in feces for over 100 years, right? I think it was in the late 1800s that somebody actually like isolated it chemically. It was only very recently that we actually like managed to isolate, well, relatively recently, we managed to isolate and sort of bank up a caprosenol producer from the mammalian gut. There is exactly one caprosenol producing isolate in culture collections that you can, order from ATCC or DSMZ, the German culture collection. And that is Eubacterium caprosenoligenes, strain HL. And the HL stands for Hog Sewage Lagoon, which is where it was isolated from. And that's just one of those, it's just a terrible phrase, right?
Hmm.
Um, like, yeah, I could have gone my whole life not knowing about the existence of a hog sewage lagoon and been quite happy. I see why nobody has tried to make a probiotic out of that one yet. Somebody did some animal experiments with it in, I think, rabbits back in the like mid 90s. And it does appear to work the way that you think it would work. And rabbits are a great model organism for this because they don't have cholesterol in their diets, right? They're vegan animals, and so they eat, mostly grasses and things like that, right? And so if you put a bunch of cholesterol in their diets, they will get a hypercholesterolemia phenotype. And what they found was that if you install some Eubacterium caprosenoligenes in them, cholesterol levels go back down. They saw a drop on par with that achieved by statins, which corresponds to, like a 60% reduction in heart attack risk in humans. Uh, so it was a little bit like, why is nobody doing this? Why is nobody working with this, right? And the answer is, partly that it's a pain in the ass to grow. And nobody had yet isolated one from the human gut. And so while I had this lab in Switzerland, while I had, some time while we were getting the consent forms worked out with legal, uh, you know, I was like, well, you can't stop me from pooping. I'll sign whatever form, so I can just work with my own fecal samples. And so I had a while to play around and try different kinds of media, right? And try and find the growth factors that and there was a protocol in literature from I think the 80s where they had isolated, reported isolating some of these bacteria from like baboons and other sources. Um, and so I tried it. I made the media, which, is a whole process that involves like, step one is go to the butcher counter, buy some brains, and throw them into a blender and freeze dry them. So really, really crazy, very mad scientist feeling. But I tried it. I made the media, and isolated one of these bacteria from my own gut, and yeah. So and did the validation with chromatography that it does produce caprosenol, and like that's sort of been the genesis of and I've been thinking about this for a long time about starting a company and sort of really rushing these things to market because there is such a need for it. But this was sort of the sign that I needed, right? Because heart disease is such a huge problem. It
Yeah.
is the number one killer of humans, over and above all the cancers put together. And if you factor in stroke, it's like hands down. Um, and it's also it's sort of the sweet spot from a regulatory perspective because in the US at least probiotics are regulated as food, right? Which is a much, much lower regulatory burden than is applied to drugs. And
Hmm.
'cause I saw both my parents were in pharma, right? And I've seen the 10-year billion-dollar process of bringing a drug bench to bedside, and it's
Yeah.
it's just a nightmare, right? There's so many things that can go wrong. And at the first hint of like an underwhelming clinical trial, like the whole thing implodes. It's bad. And so, but whereas if it's a new food, I mean, you could say technically this is a new kind of cheese, right? And if
Hmm.
you've invented a new kind of cheese, what do? You bring it to the farmer's market,
Yeah. Yeah.
and you try it, you give it to some friends, and as long as it doesn't kill anybody, you can start selling it. And once you hit a certain threshold in sales, you file with the FDA and you say, here's how we're producing this, here's blah, blah, blah. And they'll say, great, has it killed anyone? How many have you sold? Okay, that's your safety data.
Hmm.
It's a much, much lighter regulatory burden. And cholesterol in particular is great in the sense that it's very heavily precedented for what we call structure function claims in the US.
Hmm.
Because the label on any and all dietary supplements in the United States says these claims have not been evaluated by the FDA. This product is not intended to diagnose, treat, cure, prevent any disease. But you know you can make a claim about how your product will impact the structure or function of the human body, so long as your data is good and actually it validates it.
Hmm.
And so this is why, Cheerios, for instance, it's a very mid cereal, right? But it still exists in large part, I think, because it's got the heart on the box, right? The good lawyers at General Mills, the good lawyers at General Mills have gone through the process of getting, claims around dietary fiber and things like that approved with the FDA, where they can put a heart on the box that says helps reduce serum cholesterol and may lower your risk of heart attack. And so, if we can actually show that with our product, we can make a substantial impact on cholesterol levels, then we may be able to make a label claim like that, which everybody knows is, so even though we're not claiming to treat, cure, or prevent a disease, we may be able to make a claim that is sort of equivalent to that, or at
Yeah.
least that tells people what we are going to do for them.
Yeah.
And so I think it's very promising. It's exciting because there's the potential for, you know, a sort of one-time intervention, right? That has a lasting impact on people's biology. And
Hmm.
that's the thing that I think really, will differentiate this from existing probiotics in that, like, well, because it's kind of crazy. The current paradigm has us renting our gut bacteria, right?
Yeah. Yeah.
Where you have to take it every day, and that's because these are largely not human gut strains, right? Bifidobacterium, yes, it's an important part of the infant microbiome. Lactobacillus, not really. It's good for turning milk into yogurt, and it is somewhat important component of the vaginal microbiome. But like, I think 1 in 3 people have lactobacillus in their guts at baseline,
Mm.
and it looks like a lot of them it's just from yogurt.
Hmm.
So it's not really an essential component. And so, yeah, if you want to sort of keep a population of them in your gut, you kind of got to top it up every day and take them. But if we are working with an actual human gut strain, something that is native and indigenous to the healthy human GI tract and that has just sort of gone extinct because, we've been treating the internal ecosystem the way we've been treating the external one, which is to say not very carefully, then maybe we can actually just reinstall this and, like, you know, I don't know. I've had my strain for 30 something years, and I eat 3 eggs every morning and a pint of ice cream every night, and my LDL is 90 and my HDL is 90. It's good numbers.
Hmm.
And
Hmm.
so, the possibility of sharing that with people is like really exciting because wouldn't we all love to eat a pint of ice cream every night and not worry about it?
This is maybe, I think this is a related question. Are you worried that if that microbiome therapy could be too effective to be economically viable? Like so much of
Ah,
pharma loves the chronic illness that you can just sell
yes.
pills forever. How do you, what's your thoughts on how that economic model is going to work if things actually treat a condition with
if
one
it actually
treatment?
if it actually kind of cures it. Yeah, so
Yeah.
that's the I've gotten this same question from venture capitalists and stuff as I've been shopping around the company. And I'll never forget there was this British guy. He's like an angel investor. This was very early on, and I'm talking him through it, and it's just like very posh, very blue blood. And I'll never forget. He kind of like steeples his fingers and he goes, "Well, it's not a very good business model, is it? If they only have to buy it once." And just like said in that way, like in that accent, it was just so villainous. It was so
Yes.
cartoonish, of like because the wing nuts who are on about like, oh, they're suppressing our cures so that we keep buying their like
Yeah.
it's never like CEOs meeting in a smoky back room to that's the only level at which it really manifests like that
Hmm.
is the venture capitalist declining to invest because there's less money in cures than in treatments, and it's like
They'll
I,
demand you
I
insert a gene with like a really
yeah,
weird
suicide gene.
molecule that the bacteria will die unless you take that pill to keep them alive forever.
Yeah,
Ugh.
yeah. Well, I mean, and if you're doing it a drug style and you are actually like engineering the bacterium, I think the FDA requires something like that because they don't want like gene escape
Hmm.
into the wild. They say like you got to do it Jurassic Park style because in Jurassic
Yeah.
Park, or at least in the books, that's like they engineer I think like a lysine
Hmm.
dependence into the raptors or whatever it is,
Yep.
and it's well like I have my thoughts about that, which is like
It doesn't
I don't
work.
care if you engine like it's yeah, I don't care. Didn't you watch Jurassic Park? Didn't you read the end of Jurassic Park? Its life is gonna find a fucking way, guys. But like regardless, like yeah, there is that like the aspect of like one time versus recurring dosing. But you think about other, well, okay, so from a financial perspective, you think about other one-time treatments that offer sort of semi-permanent upgrades to your biology, right? Like
Hmm.
LASIK
Hmm.
or orthodontia, right? And people will pay $5,000 an eye to get LASIK, and that's just to not have to wear your contacts, right?
Hmm.
Or like $5,000 for braces to have straighter teeth, and that's purely, well, not maybe purely cosmetic, but close. And if you think about it from that perspective, it's like, all right, what if we are, yes, it is a cheese, but it is a $5,000 cheese,
Yeah,
Like there
yeah,
is
and
definitely,
it's not trivial to make.
you
You
do
need
need an anaerobic
proven
chamber or
strains,
like
yeah,
a,
handled under
yeah,
difficult conditions. So yeah, I
yeah.
think
Not
I'm hopefully you'll
not
be
all
okay.
that difficult, but like getting it to people live is gonna be like the trick.
Yeah,
And I've got thoughts on that. I'm happy to talk about it lit. But like the yeah, it's really, I think there is still I pray that there is still room in the world for a company that helps or like for something to thrive by helping everybody around it thrive.
yeah.
There is still room in this world, I think, for symbiosis because like pathogen, I'm maybe waxing a little poetic here and trying to take lessons from the bacteria on how I run this company, but there's like trying to extract as much as you can from your partner in an interaction in every transaction, like that is sort of pathogen mentality. That is sort of
Uh-huh.
parasite mentality,
Yeah.
right? But like, you think about like why is symbiosis the all-time winner as far as strategies, and it's because there is, that only works until you crash out the ecosystem, until everybody is choosing defect in the iterated prisoner's dilemma that is sort of life under capitalism, right? And the luck runs out. And
Yeah.
so, I am a firm believer in symbiosis and that the bacteria have something to teach us, and that, we can do well by, I don't know, my grandma always said do good and you'll do well, and I think that there's something pithy there.
Yeah, yeah. Now, do you just now that's only one of the many things that you have researched, and you've written these up beautifully on your Substack, and I highly recommend people. We'll drop the link at the end of the episode or in the show notes. But do you want to just quickly let people know a range of other uh,
Yeah.
mysterious chronic conditions that you think you found a possible microbiological link to?
Yes, yeah. Well, and it's, in practically every poorly understood health condition, it seems like there's a microbiome link, right? And I hesitate to draw causal connections because there are I guess I hesitate to draw causal connections where we don't have a solid mechanism because there's associations where you can see that everybody with autism has like a weird fucked up gut microbiome, but
Mm,
we know that people with autism are very picky eaters generally speaking and
mm,
and have GI issues more generally. And so there's a world where actually the GI issues come from the autism and the microbiome differences are sort of a follow-on.
mm,
But at the same time, like where there are mechanisms to explain it, it's equally valid, I guess, to postulate that the microbiome disturbances come first because we know that we are sort of ravaging the microbiome on a regular basis, right? We all take one course of antibiotics per year on average in the U.S. at least, and
wow.
that's it's wild. Because if and this is something that just is informed by the perspective of like, okay, these are actually basically human genes. But it's like if I said, okay, this pill will cure your sore throat, and there's like maybe a 10% chance that it will delete an important gene from your genome, you probably wouldn't take that pill, right? Like, I hope not, unless it's really bad. And it's like, yes, if you have syphilis or something like that, probably take the antibiotic. But
Mm,
like, if it's an ear infection, maybe see if you can do the drops, right? See
Mm.
if you can do the ear drops
Yep.
instead, and usually you can. But
And
so,
having a safety net system that if it does go horribly wrong, that there is some
oh
way back to health. That's where you come in.
yes, if yeah, and we're trying to do it strain by strain here, right? And identify the actual important microbes. I think for the desperate, because like it's going to be a while until we get all of them figured out. Like there is
Hmm.
fecal transplant, right? And the regulatory situation around that is currently pretty stupid, but nothing can stop you from doing it yourself. This is my number one message to the listeners, and this is the title of my blog is Eat Shit and Prosper.
Hmm.
Um, and it's kind of quite literal in that like if you're really desperate, if you've got Parkinson's or Alzheimer's or like one of these other diseases that like is gonna kill you, uh, it doesn't take much beyond like a pastry bag, some capsules, and a very healthy and understanding friend to try and fix it yourself.
Hmm.
And in a surprising number of cases, there are like case reports that it's working. So I mean, just to go down the list, it's like I mean, yeah, Parkinson's and Alzheimer's are both on there. As is depression, as is schizophrenia,
Hmm.
multiple sclerosis, ALS. I believe even things like celiac disease and gluten intolerance. Um, and in a lot of like, I'm not saying these are all just like dysbiosis, right? Like, because
Mm.
dysbiosis is a weird and nebulous and probably too broad category. In some of these, it seems like it's an absence of something. In others, it's probably like a presence of something that shouldn't be there, right? And so for instance, if you were trying to treat ALS with a fecal transplant, I would imagine that you'd have much better luck pre-treating with an antibiotic and then sort of paving over the ashes once you've burnt down once you've burnt down the jungle, paving it over with a square mile of healthy ecosystem from somebody who doesn't have that disease. Like that's most likely to be your sure bet. But what else do we hang on? I've gotten notes. Uh, Oh gosh, yeah. So MS, non-alcoholic fatty liver disease, ALS, type 1 diabetes, type 2 diabetes, schizophrenia, and some types of depression, peanut allergy, celiac disease, hypercholesterolemia. The list goes on.
Yeah.
But like
Yeah.
those are some of the most promising, and the ones I just rattled off are ones where I have a hunch, or like not even a hunch. I've got a theory, and like there's a molecular backing, there's a molecular mechanism. Although the type 2 diabetes thing is actually more about the shit that we're spraying onto the crops
Hmm.
than the actual microbiome. Because people have tried fecal transplant in obesity and type 2 diabetes, and it hasn't really done much. Uh, but and yeah. So I would highly encourage. And Shane, as you said, it's maybe doesn't work as well to do sort of the deep dive on each of these things via podcast, but
I,
highly
I
encourage.
I would love it, but I know we would be here for like 5 hours.
Yeah, yeah, and there's plenty of time.
Yep.
Um,
Oh, well,
so
one question I wanted to ask you on this. Do you almost worry that you have too many substantial hypotheses that
yes,
like one lifetime may not be enough?
yes. Well, and that is, I think, the only real ethical good reason to start a company, right? Is
Hmm.
if you have more than one person's lifetime's worth of work to do, and it's good work, right? Then get some other people on board, start something that even if you die, will go on and allow that work to continue. And
Yep.
so I'm really thinking about this from sort of like a generational perspective. Like let's do something that like I've when I'm in my full sort of messianic vision founder mode and pitching to investors, I've said that like what we're trying to do for biology, what like AT&T did for information, right? To make
Mm.
it sort of instantly transmissible across the country and make it available to people in a way that it just has not been to date, right?
Mm. Mm. Okay. All right. Now, are you ready to jump into some more kind of left of field questions? I've got a few of them.
Oh yes.
Okay,
Yeah,
so
absolutely.
first broad topic I put some things together to ask you is on society. And I wanted to follow your lead. You're really good at writing in a way that gets people relating and kind of tapped in. So I think most of us have had the weird experience as a child of going to a friend's house for the first time and realizing that everyone in that family smells funny, that they all smell
Yes.
different.
How's
And
smell?
that is probably at least partially tied to microbiomes. And it's got me wondering, could microbiomes have a role in kin or mate selection, like recognizing kin, selecting mates? Um, and
Oh
like,
man. You hit yes, yes. So okay, can I interrupt you here because
yeah, yeah.
uh yeah, absolutely the answer is absolutely yes. The so have you heard of the sweaty t-shirt test?
Hmm.
You're familiar with this, so
Yeah,
and
but
for the
explain it to the
listeners, I
audience.
guess. Oh, you have explained it to the audience before.
Oh, no, 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 I haven't. But yeah, do you want to
Oh,
quickly
okay, I thought you
recap
were saying.
what it was?
Yeah,
Yeah.
yeah, yeah. So sweaty t-shirt test is basically, these guys uh, took you take a bunch of dudes and you have them put on a fresh t-shirt and you have them work out for half an hour or something like that, very vigorously, get them all sweaty, and then, take those t-shirts and give them blinded to a panel of female sort of raters, right? And have them do like a sniff test and say like, okay, and some of them will go ew, right, at a given shirt, and others will say, oh, I don't smell anything at all, or yeah, that smells like a good hunky guy,
Mm,
right? And if you look at these people's genomes, you can actually sound and I don't know if this finding has been like really well validated and replicated. Don't even know what their original sample size was, so
mm,
maybe don't take this as gospel. But like basically, you can tell based on the genome whether or not somebody's going to rate a part or like rate a shirt as smelling good or smelling bad, and the determinants are in the major histocompatibility complex.
mhm,
Okay, the MHC. And this is a region of the genome that it basically helps your tissue differentiate self from other, right? And tell like, is this a human cell? Is this a, is this something I don't recognize? Is this a foreign invader potentially? Right. And so the MHC is an immune complex. And it is like I guess the finding in the sweaty t-shirt test was that people with very different MHCs uh, enjoy each other's body odor, right?
mm,
People with very similar MHCs smell pungent to one another. And this is yeah, like the hypothesis around mate selection and that is that like your body is selecting for a diverse immune system, right? Is that Mother Nature sort of wants your offspring to have a broader range of immune genes and immune recognition capabilities. Um, and like when I first found out about that finding, it was like, whoa, you can smell somebody's genome. That's fucking crazy. But when you think about it, it's like most body odor is bacterial in nature, right? Sweat itself, as it comes out the pores, does not have an odor to
mm,
it. It is the bacterial fermentation products of the fats and compounds in that other compounds in that sweat. That is what actually smells. So you can smell somebody's genome, is like really cool. You can smell somebody's armpit microbiome. Like, yeah, that's less impressive somehow when you put it that way, right? Like, yes, I know I can smell your armpit bacteria. No, but like, what that tells me is that the microbiome is determined in large part by the genome and by the major histocompatibility complex, which makes
mhm.
sense. It's not only differentiating self from other, it's probably, well, it's differentiating self from other, but you got to consider that the self is probably wider than what we, have classically considered the self, right? It's wider than the 23 chromosomes and the mitochondria. It includes the microbiome. And so,
Yeah, yeah. And the
yeah,
human
yeah.
body actively cultivates microbes that it wants to have around. It isn't just a random whatever bugs turn up, set up shop.
Yeah, yeah. Well, and so, but this is the interesting sort of duality there is that it's you know the like yes, it actively cultivates in much the same way that we cultivate a garden, right? There is you know like there are ways to sort of promote the presence of some bugs over others in things like mucin, right? Mucin is like, this is the molecule, the sort of glycoprotein that makes up most of your mucus. And not only like in your nose and face, but also like in your gut. And there's mucus lining your GI tract. And mucin's it's a beautiful molecule. It's an incredibly interesting sort of structure is function example in that it's like imagine a bottle brush, right? But each of the bristles is a smaller bottle brush. and then maybe each of the bristles on that one's another like I don't know how many layers it goes down, but like that's me. And because each of the bristles on that final layer are very hydrophilic, you can sort of see how it makes such a great water holding matrix, right?
Hmm.
And so like the thing is that those are like the thing that makes those bristles hydrophilic is sugar molecules, right? Because sugars are very hygroscopic. They will soak up water from the atmosphere and things like that. But where I'm going with this is that a lot of bacteria have the ability to eat those sugars, right? They have the ability to sort of clip one of those little brushes off and start snacking on it and free it up for their friends to snack on. And so, like there is some active cultivation in that sense, right? But a lot of the cultivation is that our body does to sort of encourage the right bacteria is done by discouraging the wrong bacteria, right? So it's like you can fertilize your garden. Uh, you cannot bring anything back from the dead, right? You need seeds. You can't get squashes out of your garden just by like putting a bunch of fertilizer down. You actually need the starter seeds, right?
Hmm.
And so your body needs something there to sort of sculpt and subtract from. And this, I guess, brings me to the other main point that yes, it's cultivation, but a large part of keeping a good garden is weeding, right? Is pulling out the things that don't need to be there, that you don't want there. I think this is where the human instinct toward dirtiness comes into play and plays an important role. Because we were talking about like how, it's not fully clear how humans get their microbiome so reliably across like because nature does not tend to leave terribly important things to chance.
Yeah.
And for a while, it looked like milk was the major vector of transmission. Like I was, this was actually a finding from that evolutionary biology class way back when in college, where the professor was talking about the milk microbiome. And it's like, oh, this is such a beautiful, elegant solution, right? Because up until 100 years ago, if you didn't breastfeed, you died, right?
Hmm.
And so you were, here's a vector for if the bacteria can somehow get transported from the gut to the, milk ducts. And deposited into the milk and fed to the infant. Here's a 100% guaranteed vector for vertical transmission across generations of these microbes. It is now like knowing what I know now and like having a really good idea of what the bacteria that you find in a human gut look like and looking at the bacterial DNA that you find in milk, it's not a lot of the same species. A lot of the ones that you find in milk are like mouth bacteria, skin bacteria, probably just like backwash from the infant's saliva kind of thing is what's going on in milk. And so that leaves sort of the question of like how are these bacteria getting there? And I really think I'm really leaning more and more towards just like this is why kids will, left to their own devices, put fucking everything in their mouths and why they will pick their noses and eat it. And this
Hmm.
is, because that is, if you think about like the house smell phenomenon of like you walk into your friend's house or whatever, you're breathing in the air, and there are, at least a couple of cells of Bacteroides from the last time they flushed the toilet and a little cloud went up, or grandma didn't like wash her hands fully before making the red bean buns. That's how you get those couple of spores into the like I guess what I'm saying is that if you think about the structure of the nose and this sort of very privileged immune surface that is the nasal mucosa, you're basically filter feeding these bacteria out of the air
I
and
love that.
then taking the booger and just inoculate, putting it down the hatch. It's like that seems like a great way to pick up a diverse microbiome that's representative of those around you, and then your body will subtract out whatever is not supposed to be there and keep the good stuff, right?
So, is it possible that some of the negative health effects of loneliness are actually coming from a missed opportunity to replenish your microbiome?
Yeah. Well, that's an interesting one. Yeah. I mean, anything is possible. I have seen the study on like I think it's like more um oh, what was it? It was like looking at like social structure in like primates of some kind of non-human primate. And they found that like the lonelier ones or the like less popular, more outcast ones or whatever had like less diverse microbiomes. But there, it's to my mind like a chicken or the egg thing, because it's, I guess like perhaps the most popular monkeys are the ones with diverse, rich microbiomes that everybody wants to hang out with, and because they're full of piss and vinegar, as the saying goes.
Hmm.
Uh, and like I don't know. So it could go both ways, but yes, there's definitely in my mind a possibility that it is like, the social isolation and sort of this like low diversity crisis go hand in hand,
Hmm.
right?
What about the other extreme? And this has got me thinking about the mouse utopia experiments, where mice that seemed to have all the food and space that they wanted reached some kind of critical density in the population where something happened to just make everything fall apart. And we're seeing to some degree similar things happening in humans, that the ecology of microbes transferring between people might have a sweet spot between complete isolation and packed in like sardines.
Hmm. Oh, so you're saying I'm not too familiar with the mice utopia experience, but basically you're saying like they reach a critical population threshold where they get too crowded
Yes,
and
yeah, that there could be a microbiome component to that phenomenon, because I don't think they quite know what the mechanism is.
interesting. I am not familiar with that one. And well, okay, so mice are tricky because they're what we call coprophagic, right?
Hmm.
They eat their own and each other's poop. And so like this is nice when you want to like if you are doing a microbiome experiment because you just leave six mice alone in a cage together and they will develop a very homogeneous microbiome across the cage,
Hmm.
generally speaking. Or maybe they'll develop a pecking order where like nobody eats Carl's shit because he's got a bad one. But maybe his will sort of normalize out if he's eating the other guys's. Point being, like, it's relatively hard. Most humans are not coprophagic,
Mm.
and it's relatively hard to extrapolate from there to what we're seeing in our world. But there is. I mean, you think about agriculture and like the conditions of like sort of the CAFOs, right? Like concentrated agricultural feed operations, where it's
Hmm.
like you got 10,000 chickens pecking around in a warehouse or something like that. Like, yeah, that's gonna lead to a population collapse. That's just begging for a virulent strain of coccidiosis to sort of take over and wipe things out, right?
Well, this is what I'm leading towards, and I think we see the same thing in our like a cultural information ecosystem, that when things
Oh,
become too large and too connected, the incentives for like pathogenic viral transmission become too high, and you don't get those feedback loops that encourage symbiosis. And we see this today with like viral online content. All it is really selected for is its ability to transmit, and it doesn't actually
yeah.
carry anything useful. And I wonder if a
Great
similar thing
way.
happens in the ecology of microbiomes, that they can be too isolated or too connected at the extremes.
Interesting. Yeah, like like and subscribe and share, and that's the only content of the video now.
Yeah,
That's
yeah.
funny. The yeah, I mean there's definitely something to that. But it's also I don't know. Well, so I actually want to bring this back around to sort of the work that I'm doing of like culturing these bacteria, getting them to humans. Which is like the difference between an anaerobe and an aerotolerant species, right? Because the reason that, you go to the CVS, you go to the drug, I don't know if you have CVS down there, but like you go to the drugstore, there's an aisle of probiotics, all
Hmm.
right? And, you look at the labels, they're all either lactobacillus or bifidobacterium, maybe a streptococcus or a yeast now and again, but for the most part, it's those two genera. And those two genera are, the existing the whole existing market because they're pretty aerotolerant. They can deal with oxygen.
Hmm.
Most of the bacteria in your gut, not, but collectively, like Lactobacillus and Bifidobacterium, typically they make up less than 1% of a healthy adult gut microbiome.
Hmm.
The other 99% are strict anaerobes, meaning they cannot tolerate anything approaching atmospheric concentrations of oxygen. When I'm working in the anaerobic chamber, we got to keep it less than 200 ppm, 200 parts per million oxygen. The air we breathe is 200,000 parts per million oxygen, right?
Hmm.
So, that presents some problems for these bacteria. But if you think about it, this is like that lack of aero tolerance is what like I guess where I was going with this is that like lacto and bifido, those are sort of the only two, not the only, but like some of the only aerotolerant bacteria that don't want to kill you, right?
Hmm.
Because E. coli is aerotolerant. So is cholera, like cholera, and like a lot of the other bacteria that are capable of living in a human gut, but are also capable of living outside it, are those are the strong pathogens. Right. And from an evolutionary perspective, if you think about the evolutionary incentives, of course, cholera wants to make you sick. It wants to give you diarrhea because that's how it gets shit out and ends up in the water supply, right?
Hmm.
And but if
Like
you
and
think,
subscribe.
yeah. Oh, yeah, yeah. But, something that dies when it leaves your body is, that is like that's sort of the relationship that you have with any of your own genes, right? Is
Hmm.
like, if I cut off a toe, it's not gonna it has no chance of going and becoming a whole other human, right?
Yeah,
And so it's like
and it's
that
got an
is
incentive to keep you alive, to keep it alive.
exactly.
Yeah.
Yes, exactly. Their only real hope at, continued existence and survival is sort of the long game of symbiosis of helping you have kids, and maybe getting farted out into their red bean buns or whatever. Like,
It,
the red bean buns has come up twice now because it's really at the top of my mind. One of my best friends growing up, this guy David, he was Chinese, and like, yeah, I just remember going over to his house, and like it was a completely unique smell
hmm.
in their house compared to my house or any of my other friends' houses. And like I probably owe a good like at least 10 strains in my microbiome to like their family or
Yeah.
something, right? And
Yeah.
this is something that I think is really cool about it is that like it is sort of like the sum of, like your the impact on your microbiome is sort of the sum of like those of the people that you are close to, that you spend a lot of time with, enough time that you like be dirty around them, that you can rip a fart in the next sleeping bag over and really like stink the room up. And that like, I don't know, something beautiful and gross there. But like it is yeah, I think I don't know. Where I was going with that is mostly just that like the from an evolutionary perspective, it's the anaerobes are the ones that are necessarily on our team, right?
Hmm.
And so if we, tried to breed like, because we could take some of these anaerobes and probably within with a couple of weeks of dedicated effort, breed an aerotolerant version, right?
Hmm.
Because bacterial evolution happens incredibly quickly. And so if we, for instance, expose them to like gradually increasing concentrations of oxygen, we might get a shelf-stable Coprococcus or Faecalibacterium. But then you might also have invented the world's first pathogenic Faecalibacterium because, as soon as it has aerotolerant, like part of the reason that we have this, can have this teeming mass of fucking virulent bacteria in our guts, uh, is that our like the entire system runs on oxygen, right? Like your
Hmm.
blood is very highly oxygenated. And so that means that your blood is a river of poison to these bacteria. So there's very little chance of them getting, sort of out of the gut and into circulation unless they have that aerotolerance
Hmm.
trait, right? But yeah, I don't know. There's something um I guess there's something there maybe to bring it back around to your question and draw the analogy. I guess there's something there about like whether whether a meme it like can, uh like whether a meme like has value in and of itself and makes and enriches your life for its place in your brain, or if it's just, sort of there to spread, if it's just in that sort of pathogenic mode of existing.
Mm.
I don't know. I'm not really sure where like what the lesson to be drawn there is, but that'll rattle around the back of my head for the next 5 weeks until it clicks into place, and I'll email you.
Now, I wanted to spend some time talking about health. And do you have any broad thoughts about the do-it-yourself microbiome community that is spreading online, the risks and opportunities and maybe missed opportunities that are unfolding around us?
Oh my gosh. Yeah. Yeah. I mean, first off, like huge respect for anybody who is ballsy or desperate enough out there to try and sort of DIY their fixes. Um it's such a deep field, it's such a complex thing, and like a lot of the solutions that are out there right now are just like they're not they don't even really begin to approach sort of fixing the problems, right? And
Mm,
this is because, yeah, right now you cannot buy a Coprococcus or a Faecalibacterium or any like, any of the Bacteroides, which is the major constituent of most people's microbiomes, right?
mm,
You cannot buy any of these at any price. And so it's like the solutions that exist. There's a great example. There's a company called Viome, right, that does gut sequencing. They'll do a microbiome sequencing, and then they have like a list of, supplements that they sort of and like they sell the supplements and stuff too, and a probiotics and stuff. And so it's funny that they'll sequence your gut, and no matter what's going on down there, somehow the solution is always like one of the strains of Lactobacillus or Bifidobacterium that they are selling, right?
mm,
Because that's what's available right now. But there's like I guess, uh, yeah, like when you do it right, by which I mean going directly to the source and
mm,
doing a fecal transplant from a healthy individual, some miraculous things are possible, right? There's a great case report from actually out of Australia of a woman who had bipolar disorder, and it was really a heavy burden on her, but had read about the connections with the microbiome, understood like that fecal transplant is remarkably safe for what it is. And
mm,
I cannot stress this enough to the listeners. Like if you look at the numbers and it is done right, or even if it's like even done right aside, if the person that is a donor is healthy and you are not like severely immunocompromised, uh, it's actually like not that bad. You would think that like eating somebody's shit is a very bad, a very risky thing. But if it is like fresh out the human body, within, and within an hour back into a human body, uh, most of the anaerobes are still there.
mm,
And, it is like what worked for that one person is likely to work for another, right? And so, this woman with bipolar did a fecal transplant from her partner, her boyfriend, her husband, or something like that, and is still in remission to this day, as far as I'm
mm.
aware.
And I think
And
there have been a small number of cases of things getting dramatically worse for people after attempting this themselves. It's not a guarantee.
yeah, no, obviously, this is not medical advice, but and I don't think like I don't think anybody listening to this is who wasn't already thinking about it is going to go out and eat shit on our advice.
Mm,
But it is like I guess I've said this on the blog that like it's black magic,
mm.
right? To do like a fecal transplant, brown magic maybe. In that like we don't know how it works, right? We don't know what all you are transferring, right? Your risk of like because we don't know what all traits are microbial. Your
Mm,
risk of male pattern baldness might be down in your microbiome. Your risk of one that we didn't throw on the list earlier of microbiome linked conditions, alopecia. The
mhm,
weird thing where like all your hair kind of falls out spontaneously. They did a fecal transplant to treat Clostridium difficile in a couple of patients in China with like in patients who also had alopecia, and sort of unexpectedly their hair started growing back,
mm, mm,
which is very strange. But like all of these things like the risk may be in the microbiome, and so but if you have Parkinson's or something like that, and you're the only person willing to shit for you has alopecia, take that trade, dude.
mm,
I would take that risk if it were me. And so there's like, to the extent that you, to the extent that these traits are microbial, you can usually see on a
mm.
person, right? Are they happy? Are they healthy? Is this somebody who, if you got them alone in the bedroom, you'd eat their ass? So maybe that's but like I don't know. This is it's a joke that's popular with it's popular with Zoomers. But there's something there. There's definitely something there. Is like the sort of subconscious cultural recognition of like we need this diversity, we need this connection.
I was
Anyways,
actually thinking of asking that question, whether changing sexual norms might be influencing microbiome exchanges.
dude, probably. I have wondered though about. I mean, yeah, like almost definitely. Um, I do think that like I don't know. I think that like
It
yes,
would be it would
absolutely,
have to
and
be pretty
maybe
inefficient for anaerobic bacteria though.
I mean yeah, probably, but again, the thing about bacteria is that all you need is one cell getting
Yeah,
in
if
there,
the
right?
conditions are right, yeah,
If the
it'll
conditions are
find a
right,
way.
yeah.
Yeah.
If there's a niche that needs to be filled or something like that, then like, hey, there's a great example of like um strains of strains of Bacteroides common to the Japanese gut, which have like a very efficient I think it's a fucosidase. It's an enzyme that breaks down seaweed polysaccharides
Oh, yeah,
very
yeah,
efficiently,
yeah.
right? And it looks like this was actually like horizontal gene transferred from a marine microbe at some point in the distant past, but it's now like 50 to 70% of Japanese people like have a bacterium that does this in their guts.
Mm,
And like if you are eating a lot of seaweed salad on your trip to Japan or something like that, like maybe that is, creates the niche that would enable one of those bacteria to sort of take root in your gut. And maybe it wouldn't persist, but it maybe it yeah, it's an interesting thing about the efficiency of it. But
mm,
like I think the I don't know, changing sexual norms is an interesting thing because hand in hand with that sort of increased transmission, potentially the increased transmission of good bacteria. There's also the potential for the increased transmission of bad bacteria,
yeah,
right,
yeah.
in the form of STIs, which is like one thing where we like there's a pretty much a one-to-one correlation between the treatable STIs and prescriptions for antibiotics,
Hmm.
right? Unless we're talking about like herpes or something like that. Like if you've got gonorrhea, chlamydia, syphilis, anything like that, you're going to be getting an antibiotic, and
Mm,
that's going to be bad news like
yeah.
99% of the time. So I don't know. It's a gamble, and I don't know. I had the sort of moment in college where like sort of realizing like why, uh, it's a moment that I think everybody has around the time of their like first STD scare, of like realizing why these cultural norms of like sexual conservatism are a thing,
Mm,
because like for a long time those were not treatable diseases. Those were things that you just were stuck with if you got.
hmm.
And the syphilis would slowly eat a hole in your brain and your face over the next like 20 years. And having like the norms of monogamy is a pretty reliable defense against that.
Hmm.
And as we've sort of invented antibiotics and condoms and things like that, like we've sort of haha, we don't need that anymore. But to the extent that we still don't have the technology to put back the things that we accidentally like, to undo the collateral damage of antibiotics, we
Mm,
kind of still like there is definitely still a lot of value in the norms of like monogamy and sexual conservatism and all that.
hmm. Hmm.
They're fit traits, right? They wouldn't be around if they weren't, especially when sex is so fun.
Yeah.
Uh,
Now, how malleable is the human microbiome? And do we pass through critical periods of flexibility and then we kind of get locked into states as the older we get?
it's a great question. It's one that I don't fully know the answer to. It's still sort of open. And like this is sort of something that I'm trying to answer in my own work here, right? Because like this cholesterol bacterium, for instance, right? Well, like we know that okay, so we know that early life is a period of that sort of flexibility and
Mm,
malleability that you're talking about because uh that's when you come out the womb, you are basically sterile inside. And a large part of the point, remember earlier, I think I mentioned this while we were recording, that like bifidobacterium is a major component of the infant gut microbiome, and it is very important in that role.
mm,
And this is because it is like, one of the best at utilizing human milk oligosaccharides, the special sugars in milk that, really sort of lay the groundwork for a healthy microbiome. And like what the I don't know, the analogy that I have made in the past is that like while you are still on milk, like while you're still exclusively breastfeeding, it's Bifidobacterium is the snow blanketing the ground, right?
Hmm.
Nothing can really get sunlight because it is just like a full sort of bifido only show. The seeds, of other bacteria, like one or two stray cells wanders in, and they, sort of end up finding their way down into one of the crypts in your gut, right? Like one of the little pockets. Like they can maybe hang out there and persist at low levels, but it's really mostly a bifido all the time show.
Mm.
Uh, Once you go off that milk and then start branching and like eating other foods, right? That's when the things that have polysaccharide degradation loci for like the seaweed eaters or whatever. Like that's when those can bloom, right?
Mm.
That is when you get onto other foods. Um, so like there is, obviously, yeah, like that flexibility early in life is a known thing, but whether it like how easy or difficult it is to make changes to the microbiome later in life without doing something like an antibiotic clean out first, it's largely unknown. And I said that, yeah, this is something that I'm trying to answer in my own work, in that it's like how much of this cholesterol. So the cholesterol reducer, we're calling it Astrobacillus. I'd say we are calling it because I get to name the genus. It's very
Hmm.
fun.
Hmm.
I'm the first person to isolate one from this genus, is previously known as, I think, like CAG 70. No, what was it? CAG 177 in the genome taxonomy database. But like how much Astrobacillus do you have to drink if you are missing caprosenol producers in your microbiome in order for it to take root,
Mm.
right? And is there sort of competition for real estate from other things? And like probably the answer depends on a lot of factors, right? Like, is your liver secreting enough bile to, or is your gallbladder secreting enough bile to support the growth of this thing, which uses cholesterol as a terminal electron acceptor? Um, are there other things in there that are, also have the potential to do that, right? But like, I would be surprised if there's not some degree of malleability, right? If like I doubt we're all locked in. I'm hesitant to draw from any of the studies because there have been some studies that have looked at like um, similarity of the microbiome between cohabitants,
Mm.
right? You study spouses versus like siblings who live apart or something like that, and you find that like the longer people live together, like the more their microbiomes tend to resemble one another. Is that evidence of person to person strain transmission happening? I don't actually know because, uh, most of the metrics that are used to analyze this, things like unifrac, sort of look at like unique. So unifrac stands for unique fraction of the tree of life, of
Mm.
like how similar are these two microbiomes. It's a complicated question to try and answer, of like how similar are these two ecosystems. The analogy I've given before is like, is a pine forest in Oregon more like a pine forest in Japan, or is it more like an oak grove next door in Oregon,
Yeah.
right?
Yeah.
And the answer sort of like depends on what you mean by like how, like what you're looking at. Are you looking at like, because obviously you're more likely to find a pine tree from that pine forest, like that exact same species and strain of pine, in the oak grove next door.
Mm.
But is that your concern, or is it like number of pine tree, or like like density average number of pine trees, or like terpene secretion, or like whatever factors that you want to define
Yeah.
as the important thing about a pine forest. And so it's like, when you look at two people's gut microbiomes that live together, and you say, oh, well, like both of these people, like this person has 60% Bacteroides and this person has like 45% Bacteroides in their gut microbiome. Uh, are they going to be the same strain? Not necessarily, but because those two people are probably eating meals together, and like the composition of your diet does, play a huge role in determining which species thrive in your gut.
Mm.
You're gonna see, similarities emerge, even if it's not like a strain transfer thing happening.
Yeah.
Uh, but we do know that, diet plays a huge role in sort of encouraging or in like which species it encourages.
Do you suspect there might be some difficult to study effects coming from very low abundance species of microbes that if you just survey like the bulk abundance of different dominant strains that you could be missing? They could be in the 0.1% that's difficult to get a consistent read on.
Yeah, I mean, well, okay, so the CAG 177, the Astrobacillus is a great example. That's like a 0.5 percenter or
Mm,
something like that in my gut. And that's like actually like that's not low, but it's not like it's not undetectable, it's not even difficult to detect. Like the standard 16S metagenomics that I use, like they give you 5,000 reads for $60.
mm,
Right? And like 5,000 reads will, that'll tell you, at least assuming like, you do a good lysis of your bugs and everything like that, like that'll tell you down to like a 0.02% abundance. So obviously not like undetectable by any means. Is it the thing that you're gonna see from like at the top of the list? If you're just looking at like the, first 100 rows in your spreadsheet? No, not necessarily.
mm,
Um, and until we, knew that GAG 177 was a caprosenol producer, it's one of those things that like would sort of coast under the radar.
mm,
At the same time, I'm a big believer in like seeing the forest and not worrying as much about the individual trees,
mhm,
to the extent that it's like, yeah, if something is like wildly out of whack, you'll see. So okay, so there's a couple of great examples here. One is like the ALS thing.
mhm,
And this is, really a hypothesis that like I just put out this post on the Substack a week or two ago. This is a hypothesis is very recent, but like a friend hit me up. Her mom has ALS, and she was like, "Is there a microbiome angle here? Is there anything we can do?" And I found a couple of case reports from I think China, and in ALS patients, they did fecal transplant. It actually helped. And they had before and after data on the microbiome. And in the before data, it's like uh, uh, both of these patients, their microbiome was about 50% Enterococcus.
mhm,
And that is an absolutely wild number.
mm,
Most people do not have Enterococcus. You find it in like one in 20 people or something like that. And then it's usually like 0.1 to 0.5% of the microbiome.
mm,
And so it's like seeing like, okay, it's 50% in these diseases. Yeah, there's definitely like a lot of times when it's like, no, it's clearly out of whack. And it turns out that Enterococcus produces hydrogen peroxide,
mhm,
which is a pretty unique trait among bacteria. And there is actually a good amount of evidence linking hydrogen peroxide to the pathology of ALS because some mutations to the human gene that produce hydrogen peroxide, like those are the genes that are linked to ALS. And it's a gain of function mutation, so it makes it a better producer. But on the other side of the coin, things like hormone metabolism,
mm,
right? Steroids are an unbelievably sensitive lever in the human body,
yeah,
and all steroids also undergo enterohepatic circulation, the same way that your cholesterol sort of gets squirted out by the liver into the gallbladder, into the intestines, and then gets reabsorbed
mhm,
after, before or after bacterial transformation. Steroids do the same thing, and you've got gut bacteria that take the sugars off of your testosterone or your estrogens, and that is what allows them back because your body sort of excretes it by putting a sugar molecule onto it, glucuronidating it, and
mhm,
then sending it out for excretion. If a bacterium deglucuronidates it, then that lets it sort of back into the mix.
mm,
And because steroids are such a like potent thing that could be active at nanomolar concentrations, um, I would not be surprised at all if yeah, like having like a 0.1 percenter of Clostridium scindens, which is a bacterium that like cleaves steroids down into like inactive or differently active metabolites. If that's something that could make a huge impact on for instance fertility.
mm,
Um, and that is not, that's not hypothetical. That's actually like sort of some data from a conference that I went to in Boston. I saw a talk by somebody who was, uh, basically walking through like determinants of fertility and like successfully carrying a pregnancy to term. And it's like if you have a bunch of Clostridium sindens in your gut, you're probably not making it. And you know,
Yeah,
so
yeah.
that like yeah, but like I guess to your point or to your question, we saw it. You know, even if it's a low abundance thing or a low prevalence thing,
Mm.
you can still see it. But you know, it's hard to know what we're looking at because so much of the microbiome is still uncharacterized, like
Yeah,
fully.
yeah. It's
We're very
it's a
lucky, but
we're in a Wild West moment. Like we've just flipped on the lights in like a dark
yeah,
forest, and there's all these creatures around us that we didn't know were there before, and we're trying to figure out what they're all doing.
yeah, yeah. Well, and that's and it's really tricky because it's like you can't even see them, or if
Mm.
you look at them, it's just they're tiny sausages, and it's like the ones that turn milk into yogurt are basically identical to the ones that give you anthrax,
Yeah,
right?
yeah. And most of them don't want to be cultured in a laboratory under easy conditions. They have
Exactly.
various demands, or sometimes known, sometimes not known.
Yeah. Well,
Mm.
so and that's to like hate to keep hitting on it, but like this is actually like the cholesterol thing is a really illustrative example because like we still don't know 100% the genes in these cholesterol reducing bacteria that are responsible for the reduction,
Hmm.
right? Like we don't necessarily know which genes carry out the conversion of cholesterol to caprosenol because like in order to identify the gene that's responsible for a trait, you got to have a lot of different organisms with that trait. And then you can do
Hmm.
knockout experiments, right? Where
Yeah.
you just like snip this one gene and see does it lose the trait. But like if that, kills the bacterium, basically like if it's its only electron acceptor that it can use, then yeah, you have a hard time. Well, and so, because we only had one of these bacteria in culture until now, that was like a known caprosenol producer. Um, it's very difficult to do the sort of process of elimination of like which of these things is not like the others and see which genes are common to all caprosenol producers. Now
Mm.
that and it's actually funny, there was a paper that came out of like Harvard lab a couple of years ago where they were like, we think we found the gene responsible, and we cloned this gene into an E. coli or something like that, and it carries out this one reaction, and they didn't show the full like basically they couldn't get it to produce caprosenol in culture, but they got it to do sort of like one part of the reaction. But
Yeah, yeah, yeah. There's normally a
you know,
cassette of genes that like push it through multiple reactions.
yes,
Yeah.
yeah. Well, and that's in particular like this is a great example of why it's so difficult is because like cholesterol's a really fucking weird substrate and
Mm.
caprosenol is an even weirder product,
Mm.
right? You think about like cholesterol forms these micelles, it's not water soluble, and the inside of most of these bacterial cells is aqueous. And so it's like I've talked to people because again this has been known about for a long time. The cholesterol caprosenol thing has been known about for 100 years. People have been trying for like 20 years to like hack whatever genes they think are responsible into lactobacillus or yeast or whatever. And I was talking to this kid who like this was his project at university for a while. And I was like, how's that going? He's like, turns out it's really fucking hard to get yeasts to take up cholesterol from the
Mm.
media, right? They don't
Yep.
even want it. And then if you did get them to do it, and then, sort of perform this intracellular conversion of cholesterol to caprosenol, now you have a fully insoluble product accumulating in the form of these crystals inside your cell, and it's
Yeah.
going to,
It's gonna
start
kill it.
poking holes.
Yeah, it's gonna kill
Exactly.
it.
Yeah. And so unless it has like the importer and exporter cassettes, as you say, like, and so this is because I've gotten the question before from people of like, well, if these things are so hard to grow, why not just hack the gene into a lactobacillus and do it there? And I've drawn the analogy to spider silk, right? Because
Hmm.
silk is supposed to be this miracle material, right? This wondrous protein that like why aren't we all wearing shirts of spider silk right now, and it's largely because like you can put the gene for silk production, like we know the protein sequence, but if you put it into an E. coli or yeast and you grow it in a vat, you just get blobs,
Mm.
right? It is actually like it has to be you need a spinneret right? You need to actually like pull it out a little strand at a time as it is produced in order to get the molecules to fold right, basically, to get the protein to like not just agglomerate into a big glob,
Mm,
and to actually form this strand. And so some people have actually had luck recently hacking these spider silk genes into silkworms because they already have that whole extrusion process down.
mm.
But like, at a certain point, well, like, I've made the joke that at a certain point, yeah, you're better off just breeding spiders. But like, these people have found like actually the smart way to do that, which is take something else which makes silk and teach it to make spider silk. But
Mm.
anyways,
Now, you're very focused on the huge target of the gut microbiome, but do you have any thoughts about other potential microbiomes? So I know there's talk about a brain microbiome, that our brains may not be as
ah,
sterile as we thought.
that's
Tumors have
cool.
their own microbiome for cancer. The mouth has a really distinct microbiome as well. The people are starting to make moves on engineering different strains to prevent cavities. Like, what do you think about
Yes,
all of
yeah,
that
lantern.
other area?
Yeah, lantern's a really cool one. The oral microbiome thing I think is particularly promising. I think it's really cool, and I'm delighted to see that they're like sort of bringing that to market and taking that same sort of like cowboy style approach of like we're just gonna put this out there and see if it helps people.
Mm.
Um, the brain microbiome, absolute lie, absolute myth. This is and some researchers will probably get very mad at me for saying this, but there's the old like I as soon as I saw because that was a headline like article in Science
Yeah,
not too
yeah.
long ago. It was like the brain has its own microbiome. And I just like as soon as I saw that one, I put my face in my hands and I was like, oh, because like there's that old thing about like a lie has made its way halfway around the world before the truth has its
Yep.
pants on.
Yep. Yep.
And like I know that 5 years from now, I'm still going to be meeting people who are like laboring under the delusion that there is a brain microbiome because they were in an airport and they saw that cover and didn't read the article or anything like that. But like that, oh yeah, no. Listen, if you have any substantial quantity of bacteria in your brain, that's a problem. That
Yep.
is called meningitis.
Yeah.
And there is like I read the paper, I read the materials and methods, and it's just you know it's very like you think about like the work that we do when we're culturing. Like you gotta have sterile materials, you gotta work inside a biosafety cabinet and everything like that. Um, but like that is nothing compared to doing sort of like trying to do a proper experiment where you look at like whether there are bacterial cells in the DNA or in the brain, or
Mm.
whether there's bacterial DNA in the brain of a deceased human. Because not only do you have to keep things like sterile where there are like no live bacteria, you have to keep things DNA free.
Hmm.
And so like that brain is located inside a skull, which is located underneath a bunch of skin. And that skin is covered in bacteria.
Hmm.
And even if you kill, like you alcohol and sanitize and iodine the fuck out of that skin and kill all the bacteria on it, their DNA is still there.
Yeah.
And
Yeah.
when you cut that skin open and that skull open with a buzz saw, which I don't know how else you're going to get it open, it's gonna go flying. It's gonna go
Mm.
fucking everywhere. And so like if you get a few stray cells of Streptococcus or Staphylococcus, more likely Staphylococcus, from that skin in the wound or in the tissue, like it's just so hard to avoid that kind of contamination.
Mm.
And the fact that most of the bacteria that they found in those brain samples were skin bacteria,
Yeah. Yeah.
I'm like, I'm sorry, nah. There is like, and it's a very important point. Well, not that important, but it's a point about the definition of the word microbiome, right?
Hmm.
Because like there's, like there is no bloodstream microbiome because if you have bacteria in your blood, that's like sepsis, right? Like that is an infection. A
Mm.
microbiome is typically like an ecosystem, right? It is like there's ongoing metabolism. It can be part of a healthy body. It's like part of a normal system. And there's, interactions going on among the bacteria and things like that, right? It's not an overtly pathogenic condition, but like
Hmm.
yeah. And so that's sort of I don't know. And the tumor stuff, it also looks like a lot of that. I mean, in some cases, I think I like it seems likely if we're talking about like the colorectal cancer
Mm.
tumor microbiome or the prostate cancer tumor microbiome. Yeah, sure. Those are right down there in the ass next to all the bacteria. There's probably like bacteria doing tissue invasion and maybe even inducing the formation of tumors.
Mm.
Uh, in yeah, in other cancers and things, like a lot of those signals it looks like are not fully replicating. And I'll never forget when I was at CF at the Swiss Institute, somebody was doing like a blood microbiome sort of project and like looking for a blood microbiome. And he's showing his results in a lab meeting, and I had to raise my hand and be like, I hate to say this, but these, we're seeing Cooper avidus and a couple of these other like microbes on there, which are like Ralstonia was one, and it's like these are classic what they call kitome, right? Like this is the
Oh, yeah.
microbiome of the factory that produces the sampling kits,
Yeah.
right?
Yeah. Yeah.
Residual bacterial DNA in the water supply there and stuff like that, and then other things showing up on his slide were like brassicaceae, and I'm like, that's cabbage. Just saying that's
There's,
cabbage.
there's a
And
cabbage in
I
your
was like,
blood.
and he kind of like looks at the slides, and everyone's like, oh, okay. I hate to be that asshole, but like, guys, like look twice at it before you bring it to meeting.
This is a
Um,
related question. Do you worry that particularly academic science is becoming too polite, too deferential, that the culture has lost its
oh,
willingness to call people out, where you're like, I think your life's work is bullshit. You might want to do
bro.
something else.
Uh, yeah. So I had a paper, or I have blog post in drafts that might stay in drafts because it is some of this impoliteness. Like yes, I 100,000% think that academic culture has become too polite. Nobody's willing to have beef anymore. Journals are not even willing to publish beef anymore, which is a
Mm,
shame
mm,
because I long for the days when you could like follow a volley of scientific, retorts back and forth between two researchers who were like, "Oh, well, your work is bullshit." But so there's a study that came out a couple of months. Let's see, I was in Amsterdam at the time, so this would have been like October of last year about the vaginal microbiome and some interesting sort of chemicals that were identified in vaginal wash sort of samples and the potential antimicrobial effects of these compounds. Basically, they were saying like, okay, we found these beta carbolines in the vaginal rinsate. And they seem to have antimicrobial effect, and we like grew some lactobacillus in culture, and did our chromatography and we found these same beta carbolines in there. It's all bullshit. 100% bullshit. And this was like a professor who it was her first published paper after getting her lab at Harvard like off the ground. And it was like she was originally supposed to be studying something different and COVID hit and so they pivoted and ended up doing vaginal microbiome work. And I wouldn't say this unless I were 100% confident and had already taken it to the journal and then been turned down because they were like, we just don't want to touch the controversy there. But like, I went into their materials and methods, went into their supplemental data. And so, point of order, beta carbolines are a very interesting class of compound. This is like what you get when tryptophan can react with like aldehydes and basically cyclize and form like a three ring circus. And basically, uh, I saw that this paper was about beta carbolines produced by the microbiome, and my hackles immediately went up because neuropsychiatry spent like a decade chasing beta carbolines
mm.
in I think the 70s around the invention of HPLC because it was like suddenly like sort of out of nowhere this really strange and interesting class of compounds appeared in like mammalian brain samples. And they're very interesting because the tryptophan derived beta carbolines are like MAOIs. A lot of them act as monoamine oxidase inhibitors, which is a class of drug that's used in antidepressant and sort of increases the concentrations of serotonin and dopamine and things like that. And they were found at like elevated levels in alcoholics. And so everyone was very very interested, very curious, and a lot of people spent time chasing down these leads, and a lot of the findings like didn't replicate, or they would get wildly variable results from lab to lab. And it turns out that like partly because serotonin and dopamine, things like that, are present at such small concentrations relative to the volume of brain tissue that you're like sampling. The residual aldehydes in like the hexane or like whatever extraction solvents that you use for
Hmm.
HPLC was enough to like react with the serotonin and things and create these beta carbolines. It was an artifact,
Yeah.
basically.
Yeah.
And I only knew about this because, I had this book, Tetrahydroisoquinolines and Beta Carbolines. It was like conference proceedings that I got from my dad from like, it's an ancient tome from like 1978. And yeah, like it's just like the epic saga of like all of these scientists like trying to figure this out and basically concluding that a lot of this was noise, right?
Yeah.
And like brought my concerns to this because my hackles went up. I opened the supplementals of this paper, and they had the samples sort of like that they detected these beta carbolines in vaginal wash, and had it sorted by Nugent score, which is a an indicator of bacterial vaginosis. That's sort of like the sniff test basically of like 0 to 6, and 6 is really bad or something like
Mm.
that. And they had it sorted by Nugent score, and but I noticed like there's wildly variable levels of this compound in the vaginal rinsates, and I noticed an interesting pattern, like something looked a little off, and sort of filtered and sorted their table by sample number, right? Like by sample index of like this was sample one, this was sample two. And if you do that, it's just a straight concentration decreasing with sample number, right?
Mm.
Like rather than, in their paper, they report that, oh, it's associated with Nugent score. Uh, and, it's like a are like a correlation coefficient of like I don't know, like 0.2 or something like that. If you sort it by sample number, the correlation coefficient between the sample number and the amount of these compounds was like 0.9 or something like that. It is 100,000% artifact,
Mm.
right? Like the kind of thing where it's like sample bleed from using a non-heavy standard in your chromatography column. I sorry to get way down into the weeds here, but long story short, it's the kind of thing that like if you know what you're looking at, it's so obvious. It's painfully obvious. And I brought this to the researcher and with like emailed her with the subject line like sorry. But like and obviously like you can't own up on something like that when it's like your first paper, you're new tenured faculty or like new tenure track faculty and you've got your lab up and running. Like
Mm.
you don't want a retraction on your first big paper.
Yeah. Yeah.
And, the journal doesn't want to touch it. They don't want to issue that retraction because like that's controversy. That's could be seen as I don't know. uh, I don't even really understand the
It's a sad
I,
situation where everybody loses if they tell the truth.
yeah. Yeah, and so I've definitely I don't know, I'm a little bit black pilled on academia generally
Mm.
because it's like there is so little um I don't know. It's easy to put out findings that work and seem plausible, but aren't real because you're counting on, like having parlayed that prestige that it creates into like grant funding or tenure or whatever by the time anybody actually like tries to replicate your findings,
Yeah.
right? Or like tries to put them into practice in the real world. And it's because, as you said, like industry and academia have sort of both been very flat-footed on the microbiome that like that gap has gone so long and that so many things have sort of like emerged that like have gone untested and have let that gap sort of widen.
Mm.
Sorry, I'm rambling now, but
It's okay.
next
Um,
question.
well, one, so one, I don't know if you picked up, but the frame of the work I do in this podcast is that all of the complexity of our current industrial technology may not be sustainable in the long run. And
Mm.
whether you agree with that or not is up to you. But I'm curious if you have any thoughts about how microbiome focused therapy could continue without genomic sequences, without supercomputers and databases, and maybe even without particularly advanced culturing techniques. And even in the short term, do you think that might be promising for microbiome-based therapy outside of the developed world? That it's a technique that could work in
Mm.
places with fewer resources?
Yeah. Yeah. Well, and this is, I honestly thought like, sort of struggling with the regulatory environment in the US, which treats poop as a drug and does not seem to have room for something that just like works and doesn't need a double-blind randomized placebo-controlled trial to prove it, like a blood transfusion sort of, that like I've thought about like, man, if I could just go to Cuba, like this would take off,
Mm,
right? Of like an environment where it's like the focus is actually on like helping people rather than making profit because it's all on the government dime anyway, and the government doesn't have a lot of dimes to spare. Like it's gonna, that's where the incentives, I guess, are more aligned with like a natural solutions like this. And yeah, so I think it's hugely promising. I don't, I mean. Yes, I think it's hugely promising, especially like in terms of things like fecal transplant, right? Again, that's a hard sell for a lot of folks, but it is, I guess, like a lot of that is cultural,
mm,
and if that Overton window can be moved, and like nothing moves the Overton window like miracles, like if there are people with MS or whatever who like come out of that who via a fecal transplant, then like that's pretty solid, especially in places where I mean, first world, third world, we don't have treatment, we don't have good treatments for MS, regardless of like whether it's, whether you're a wealthy country or not. But
mm. The
as
only
yeah,
thing I could see working against you is that a lot of the conditions that microbiome therapy is really good at treating are first world diseases because we've messed up our microbiome so much. And if you go
yeah,
to really undeveloped places, they don't, the incidence of those diseases is very low because they
oh
don't
yeah.
get dosed with antibiotics on a regular basis.
You would think, yeah. A lot of places it's surprising though that like because like I don't know, you go to like Ghana or something like that, like you can probably find antibiotics just like over the counter at the drugstore,
Hmm.
uh, in a way that now you can't necessarily here because there is like a level of stewardship now.
Mm,
But no, your point is well taken that like when I've talked to, people from Nigeria and things like that, like nobody has allergies there,
mm,
right? Like and part of that is probably I mean there's a variety of things going on, but yeah, no, I definitely it's a great question. And I read a little bit of your stuff about like how sort of the wheel of history is has turned on, the couple of species that we domesticated back in the cradle of civilization, right?
mm,
Uh and like a couple of species of wheat and grains and animals. Uh and I really think that yeah, like the future will be determined in large part by like how like whether we can get these the internal ecosystem domesticated in a similar way and get some Bacteroides out on the market and get them approved.
mm,
But like to bring it back, I in terms of like low tech solutions and ways to do this without like genomic sequencing, like I am trying to find those right now. I'm really working on like ways to quality control a ferment um without having and maybe we'll use molecular techniques, right? But like it doesn't have to be expensive. It doesn't have to like a thermal cycler to do PCR and gel electrophoresis rig to do
mm,
agarose gels.
mm.
Not that hard, not that expensive. And so do you remember like have you ever seen like old crime shows like CSI kind of stuff where it's like
Mm.
there's they're doing a DNA analysis where they like hold up they're holding up a sheet and it's got like bands on
Yeah, the
it,
old
right?
restriction analysis.
Yes, yeah, exactly. It's like we could, like if you do that kind of restriction enzyme analysis on a bacterial genome, it will tell you like whether or not like I'm trying to develop that right now as a QC for the astrobacillus for the cholesterol reducer because like there's a world where it only works if it's fresh,
Mm,
right? Like because freezing things you can do a pretty good job of cryopreserving things with like sugar and glycerin, but like you know it's hard to beat something going in still alive and metabolically active like when it goes down the hatch and like you know that means that if you want like to because quality control in microbiome or like probiotic manufacturing usually involves like plating the bacteria and letting them grow for a week and then making sure that the only things on your plate are the things that are supposed to be there.
mm,
You can't really do that and still have it fresh and the same thing that it was when you plated it. So
mm,
like, I'm working on like can we do like restriction enzyme analysis and like compare to a known standard of like the pure bacterial DNA and say like, okay, this is the only thing in there, right? And so it's like you can imagine like similar approaches used to yeah, like build out like a enzyme library or like a build out like a what do you call it? Yeah, just like a standards library of like, okay, this is what like a bacterial like what a Bacteroides chops up into. This is what like a Faecalibacterium chops up into, or an E. coli. And so, I don't know. There's definitely infinite ways that a more creative and more knowledgeable person than myself could probably find to do this low tech, right? And the same is true with like the actual growing of these things. People say, oh, anaerobes are hard, anaerobes are impossible, and this is why the field is not advanced. But it's like in the 70s, 80s, they were doing this without anaerobic chambers. They were doing it with just like a tank of oxygen-free nitrogen and test tubes that, like with rubber stoppers,
mm.
right? You got to autoclave the test tubes sealed, but it actually you can do that, it turns out. They say you're not supposed to, but like if you get thick enough glass, it actually does work, and then you don't need the chamber. I,
Yeah.
I had to go up to the manager of the lab facility that I'm working in the other day, and be like, hey, so. I am trying something in the autoclave, just FYI. Like the flasks are rated for this pressure, and I don't see any reason why they wouldn't work as advertised, but like you're
Just in
really not
case
supposed
you
to
just in case you hear something go off.
yeah, exactly. I'm like wearing the face shield. I'm like in the next room to the autoclave, just like listening for a bang. But hey, worked out great. And like I don't know. This is because we talked earlier a little bit about like the sort of cowboy attitude or mentality, and like it really, uh, I think like the rate limiting factor on our progress on this front, which I believe is a hugely important front. All the evidence suggests that like our like once we can manipulate the microbiome with enough finesse, like it's fully a step up in our technological capabilities as a civilization and like quality of life for our species, the same way that like figuring out all the vitamins was,
Yeah.
right? Like, and I believe that the rate limiting step or the rate limiting factor in our progress on this front is balls, is just like the willingness to try things that you are maybe not supposed to do and that you're maybe gonna like blow something up or like poison yourself.
Hmm.
But like because the astrobacillus is a great example of that. I have a family friend who is he's run a successful drug company before. He was like, oh well, why haven't you done animal work? And you could really do some animal work. And it's like I've already drank the bacterium, Steve. Like, and when I told my colleagues at CF, like, I have tried this, it seems to be safe. I remember one of them like raises her hand. She goes, what are you talking about? This is, Steven, this is Rick behavior. And I was like, this is what? And she goes, Rick, it's like this cartoon, Rick and Morty. Oh, cool. The highest compliment you could have paid me.
Um,
Like to be, drinking the serum down in the lab. Like it is, but because this is a big part of the reason that Lactobacillus and Bifidobacterium are the only probiotics that you can buy is just that they have a history of use and that they
mm,
seem to be safe.
hmm.
In that, but like and so it takes somebody being the first guy to drink it. And yes, you can do mouse work, and that will give you some degree of additional confidence, but it's I think equally likely to lead you astray because it's like if these are human gut bacteria, What if the mouse's immune system recognizes it as a pathogen where humans would not?
Mm,
You know, like,
mm,
and at the end of the day, we're interested in how this thing is going to behave in people, and so like somebody's got to just fucking do it. And
mhm.
obviously, like, it being a strain isolated from my own microbiome, I was like, it makes the most sense, right, to do, and it's probably the most, I don't know. My outraged colleague at CF was like, what about the ethics approvals and the animal work? And it's like, ethics is not what some guy on a committee in Zurich says it is. It is like, can you sleep at night with the work that you are doing and more importantly, not doing? Because,
Mhm.
you know, there is the possibility of harm if you take a drastic action. But again, heart disease, number one killer of humans, guys. Like
Yeah.
people are dying. Somebody's dying every 1.2 seconds. And that's a certainty of harm that if we do not act. So
Mm.
I don't know.
Now, moving on, I want to think about the implications of this for evolution, but we can start. Do you have any advice for people who are breeding plants or animals, how to include consideration of the
Oh,
microbiome into the work that they do? Because
oh, that's a
every organism has its own microbiome.
yeah, yeah. That's a great question. I think I mean, yeah, I would say that okay, so breeding animals is a great one. Because there's a substantial amount of work showing, for instance, that like the lipid profile of uh like dairy cows. And this is actually a paper that I am technically an author on. I insisted that they not put me on it, but like that because I'm literally just doing English language editing for these Turks at CF.
Mm,
But that's the culture there. So I'm technically an author on a paper about like the like lipid profile in like uh Jersey cows or something like that. Anyways, like a lot of it microbiome dependent, right?
mm,
Like where if and so I guess if somebody is trying to improve like the yield or like lipid profile in their dairy cows or something like that, like rather than assuming that it's all in the genome and that you need to like do another generation of cow, just try a poo transplant.
mm,
Just take your best, like take the cow that is the most, like that has the best profile on the trait that you want, and like just try it. Just see if the trait is transmissible to another animal
mhm.
by transferring the microbiome, because so many of these traits are microbial in origin, and we just like don't even know yet. Um,
I
and
have very
that
strong suspicions that when plant breeders do variety trials, they'll often plant 20 different strains from all over the world in the same bed, side by side. And we
Mhm.
now know that seeds carry microbes with them to some degree. I suspect that
Mm.
part of what is happening in those trials is that microbes are being exchanged between all of those different strains of plants,
Uh,
and you end up with some kind of complex mixture that isn't just the nuclear DNA being shuffled in subsequent
Interesting.
generations.
Yeah. How close do, well, I was gonna say how close do plants have to be for that kind of like exchange to occur, but it's probably hard to say, right?
Yeah, yeah, nobody really, and again, it's another one of these frontiers that nobody, like, we've only just realized this is a factor. So, yeah,
The stuff around like, managing soil microbiome for yeah, to improve yields and stuff is well known and well understood, I think. But like
mm,
um I do think that there are I think there's a lot of sort of dark matter there in terms of like terroir, like the things that and I think the French would like crucify me if I like if they would crucify anybody who actually like managed to make this work. But like you could probably I suspect you could like take soil microbiome from, some of the old vineyards in France or whatever and like move it to California and probably get,
mm,
like you might be able to get a French tasting grape for your wine like out here to whatever extent people can actually like differentiate among those things. I
mm,
think a lot of the like terpene profile is probably going to come down to like yeah, weird and unexpected things in
in
the
the
soil.
microbiome. Yep.
Yeah,
I
and
have it.
maybe some of those wouldn't transfer. Maybe they wouldn't support with the mineral profile that you've got out
Yeah,
here.
yeah.
But
I have a related question. This is kind of a tricky one, but human beings radically changed our diet during like a key turning point in our evolution, and probably we changed our microbiome at the same time. And I'm wondering if you have any speculations about how that might have changed human evolution, that it wasn't just the human genes involved, that really core part of our metabolic profile had greater effects than we currently estimate.
Hmm. Well, so when you say that, you're talking about like the shift to like a you're talking about like the shift from hunter-gatherer style
Well,
eating to like
that was
agricultural
one big
style
shift, but
or
probably the bigger one was to cooked food that goes right back to
Hmm.
the root of the genus Homo.
Oh, well, I think that's probably like the speciating. I mean, that's like one of the speciating differences probably is that because like when, and obviously like the stomach acid does a pretty good job, but you think about like buzzards, all right?
Mm,
Like or other like carrion eating animals. Even like I think birds generally actually, uh, they have gut microbiomes that are largely determined by like their diet, right? Like where it's like fully like whatever bacteria were in the thing that they have eaten, like that's what ends up being like pooped out as part of their gut microbiome.
mm,
Um, and if you think about like by reducing the input load, like the input microbial load, it enables a much more stable gut microbiome,
mm,
right? It enables the human gut microbiome to sort of like lock in and stay as it is generation to generation without too much external influence, right? Is suddenly everything going in is just feeding the bacteria that are already there rather than introduce, and it's sort of like a maybe that's like a it maybe that's almost halting the incorporation of new things into the microbiome in some way, right? Like you wouldn't expect because obviously like I mentioned that a lot of the species that you find in the human gut are like have been there since Homo neanderthalensis days and probably even before that divergence. But like where do those come from, right? They
mm.
must have been environmental at some point and like made their way in and sort of found a happy home in the ecosystem of the human gut. But like and there is still horizontal gene transfer happening from transients like with the Bacteroides that learned to eat seaweed from
Hmm.
something living on seaweed, but like probably not as many fully new genera just like or new kinds of things just like entering the microbiome, finding it amenable, contributing their, like perhaps contributing a benefit, and making their home there, right? Like that's
Here's a really crazy idea. There's a
hmm,
mystery in human evolution that modern humans, Homo sapiens, were dramatically more um, creative and variable. Like they've got a whole lot of weird behavioral traits that kind of came from nowhere. And your work on things like schizophrenia possibly being linked to gut microbiome, there's a remote possibility that that transformation in behavioral traits was actually driven by a microbiome change. And that was made easier because of our cooked food, that our gut microbiome is more disconnected from the
from the
surrounding
environment.
environment.
And yeah, there's definitely the potential for something that I don't know about connecting it to the schizophrenia thing as much, but like well, huh. No, I like that. I definitely there's like when does this transition occur? This like this behavioral Cambrian explosion kind of thing happen,
It's about 50,000 years ago and it happened really suddenly.
and
And
around the time we invented cooked food.
no. It's further. Cooked food's way further back. The 50,000
Ah,
years ago, behaviorally modern human transformation happened very dramatically, where suddenly there's art and scribbling and tinkering, and a kind of creative obsessiveness that seems to come out of nowhere.
huh. That's very interesting. I mean, is that around like invention of like complex language and like symbolic language, or like how far back is that? Because I don't know jack shit about evolutionary history,
Yeah,
you
the
know.
the language part they're not as sure about, but definitely if you
Mhm.
just looking at artifacts, they go from extremely plain with just a small amount of ornamentation to being suddenly a huge amount of ornamentation.
Hmm. That's interesting.
Mm. It's a
I
very
have
provocative theory and completely unsupported, but it kind of fits what we know
no,
about
listen,
modern.
man,
Yeah.
that's anytime I'm at a conference and I hear a researcher go, now I have no data to support this, but that's when you know you're about to hear the good shit. That's when
Yes.
about to get the like thing that is going to be like orthodoxy in 100 years.
Mm.
So like that's
Well,
pretty cool. I like that. But like, there's definitely
well, it's like the stoned ape hypothesis that hallucinogens were
yes,
really important
yeah,
for
yeah,
cultural
yeah. That
transformation,
psilocybin
but the
and
hallucinogens are
shrooms.
coming from inside the gut.
Yeah. Well, okay. And so I'm gonna step it down one because like I do think that okay, so and it's worth going into the schizophrenia one really quick because it
Mm,
is, for readers who are or listeners who are not aware, and I can't imagine they would be because
mm,
it's literally just me espousing this basically. But it's a relatively quick one and worth getting to the finer points on.
mm,
So
mm,
uh there are yeah, so in about 2023, a study came out doing a proper look at the gut microbiome and schizophrenia, like actual shotgun metagenomics, where you look at the genes of all the things that are down there. And what they found was, and this is a well-powered study. This is like 100 people, and like, a third of them controls. And what they found was that schizophrenics had, on average, like, there were some severe microbiome disruptions. And the schizophrenics have, on average, like 40 times as much of a bacterium called Ruminococcus navis as controls. And, there's a couple of other things dysregulated, a number of other bacteria that were like similarly upregulated. Um, and the authors of this paper, they sort of just concluded that like, well, it's correlated very tightly with medication dosage, and so probably this is the drugs. This is clozapine and other antipsychotics induce changes in the microbiome because we know that they cause constipation and things like that. And that's that. Nothing to see here. Kind of move along. And but
mm,
when I saw this paper and I opened up their supplementals because the good shit's always in the supplementals, I was flabbergasted because when I was working back at Holobiome, we actually had a whole program around Ruminococcus navis and similar bacteria. And a number of the other bacteria that we were looking into, they are in the supplementals. They are on this table as being significantly elevated in schizophrenia, like almost as much as navis in some subset of the population. And the thing about Ruminococcus navis, the really fascinating thing about this bacterium is it produces tryptamine and phenethylamine from
mm-hmm,
tryptophan and phenylalanine, which are dietary amino acids. And now tryptamine is the core structure at the like base of most psychedelics, right? Psilocybin, psilocin, LSD is like kind of a tryptamine structure, but like a lot of these things, DMT, right, dimethyltryptamine,
mm-hmm,
are quite structurally similar. And then phenethylamine is the, structure at the root of the rest of the psychedelics,
mm.
basically. The 2C series, amphetamines, MDMA, which I know amphetamines maybe not classical psychedelics or anything, and maybe MDMA isn't either, but definitely like potent psychoactive drugs,
Yeah,
right?
they'll be pushing buttons somewhere in your brain.
Certainly.
Any
Well,
related,
but
any,
the thing is that,
yeah.
phenethylamine is found in like chocolate, it's found in the diet, and ordinarily when you eat it, it doesn't do much to you because it's broken down by monoamine oxidase in your liver on the way into circulation, right? It usually does not even make it into the bloodstream.
Mm,
Um, the interesting thing about gut bacteria is that, they are secreting metabolites in your colon and rectum, which skips first pass metabolism, right? That
mhm.
actually bypasses the liver and goes directly into the bloodstream. So it's much more akin to smoking or injecting a compound than to eating it. On top of that, well, okay, backing it up. Somebody has done a study where they, and this was, I think, around the same time actually. Like they did a study where they monocolonized mice with Ruminococcus navis, and they found that it does elevate levels of phenethylamine in the bloodstream and in the brain, and that especially like if you like sort of damage their livers. Um, And what happens is that in response, because like phenethylamine is again it's the core structure of the base of amphetamines, right? So it will have similar effects. It's stimulative or stimulatory, like a sympathomimetic, I think is the word, and it'll keep you awake at night. And what they found is in animals, if you monocolonize them with Ruminococcus navis, uh, their brain, the chemical, the phenethylamine that crosses the blood-brain barrier induces sort of neuroinflammation, right? The brain is not happy about the phenethylamine being in there, and so what it does is, in response, it upregulates monoamine oxidase, and it
Mm.
upregulates the enzyme that breaks phenethylamine down and destroys it. This does a pretty good job of clearing out phenethylamine, but it also, in the process, clears out serotonin, dopamine, and norepinephrine. And those are sort of the antidepressant three, right? Those are
Hmm.
the cocaine three, right? You need a lot of them if you want to have creative thoughts and be happy and feel like a person, and learn things rapidly and all that good stuff.
Mm.
Um, and so, you would expect from an excess of phenethylamine in your bloodstream produced by your microbiome, you would expect a depression-like phenotype to emerge, right? Because the brain is responding to that phenethylamine by upregulating MAO and breaking it down, but also breaking down your monoamine neurotransmitters. Now, the other, and this was not even in the post that I put up on schizophrenia. So basically, like, the long story short of the hypothesis is that it's actually quite a solid mechanistic fit that, Ruminococcus navis and similar bacteria being elevated in schizophrenia like could actually be causative and driving the disease because, a depressive like phenotype is most of schizophrenia, right? The
Yeah,
the, the
yeah, the
classic
negative
like
symptoms.
thank you. Yes. Yeah, exactly. The classic, delusions, hallucinations, that's really like a rare-ish, episodic kind of thing, or at least it's like, yeah,
It
it's
comes and
not
goes in
what the
cycles,
day-to-day
which
life.
probably, well, to extend your
Maybe
theory,
you're
that
living
might
with.
relate to fluctuations in the microbiome.
Yeah. Well, and so this is so the bit that's sort of like not even really in that post is actually to tie it back that uh, like you do a dive on the supplemental materials on this paper. They did what we call a pathway analysis of the microbiome, right? Where you actually like looking at what genes are there, not just the bacterial species names or anything like that, but what genes are there and active in this ecosystem. And what they found, the most significantly upregulated gene pathway was ethanol degradation. Uh, and that to me was very interesting. And like there are a number of bacteria that produce ethanol, right? In like from glucose and things like that. Ruminococcus navis is actually one of them. It can ferment glucose into alcohol, but it then takes that alcohol and it breaks it down further into acetate, right? Acetic acid vinegar, right? This is the process of making wine, and then vinegar is breaking the glucose down into ethanol and then into acetate. Uh, the intermediate there is acetaldehyde, right?
Hmm.
The process of breaking the glucose down into ethanol also releases some acetaldehyde, and then the process of breaking the ethanol down into acetate, it stops at acetaldehyde along the way. And as we discussed earlier, if you have aldehydes and tryptamine, or phenethylamine, well, tryptamine specifically, uh, the aldehyde can react with the amine and then it'll cyclize and you get a beta carboline. You get an MAOI, right? You
Hmm.
get a class of compound that we know for a fact inhibits monoamine oxidase at quite potent, like it's quite
Uh,
potent at micromolar, nanomolar concentrations.
hmm.
Point being that like you would if you have, periodic ethanol, like periodic surges of ethanol production and degradation in the microbiome, in combination with this sort of more constant tryptamine, phenethylamine production, you would have breakthrough periods where it's like now you've got not only this sort of psychedelic or like you've got not only this thing that is causing your brain to upregulate the MAO in order to sort of defend itself against these weird exogenous psychedelics. Now you've got the MAOI that will like disable that enzyme and let them, it's like I've compared it to like the tryptamine, the phenethylamine, it's like you're trying to listen to a symphony on the radio and there's static on the line, right? That static is the noise, is the phenethylamines or whatever, that would that's sort of like blocking out your signal or making it hard to hear that symphony of, and by symphony I'm referring to like all the you know the dopamine firing that is supposed to sort of like govern our learning and behavior and reward system and all of
Hmm.
that.
Hmm.
And so, if there's static on the line, you turn down the dial, right? So that it doesn't blow your fucking ears out. But then you're missing the strings, right? You're
Hmm.
missing the subtler parts of that broadcast. And, that is sort of like the depressive like phenotype, the flat affect, right?
Hmm.
Is turning down the overall volume by upregulating the MAOI. The beta carbolines, the intro or like I'm sorry, the upregulating MAO. Beta carbolines introducing an MAOI periodically is like your friend grabbing the fucking volume knob and cranking it all the way back up,
Hmm.
and you're blasting your ears out with static, and that is sort of like what you would expect. I don't know. You would expect that to lead to a syndrome a lot like the positive symptoms of
Yeah.
schizophrenia, the
Yeah.
delusions, the hallucinations, the manic episode, the you're not sleeping and the government is sending you signals through your teeth or
Mm.
whatever.
Mm.
And I. I guess where I'm going with this is that, like I'm inclined to view the processes going on here as like inherently pathological. Like, and maybe it is like good and fit to have some small level of these because Rhamnococcus navis is found in about 50% of people's gut microbiomes. Those people are about 30x the odds ratio of cardiovascular disease and heart disease. But it's usually present at like 0.1% of the microbiome, something
Mm.
like that, right? It's a very small amount. And maybe, yeah, maybe if you've got like 40 milligrams of phenethylamine producing cells in your day, you're a harder worker or you're like more athletic and a longer runner than you would be otherwise. Um, I do actually, if you look at like where this bacterium is abundant, it's a lot of the Nordic countries and stuff like that, and then also
Ah.
Japan. But like,
Hmm.
yeah, mostly Nordic countries like Russia and Estonia, but like those are all sort of like colonized by like Viking kind of descent. I don't know. It just does track with like the Viking bloodlust bacteria, like being like the trait being microbial in origin, and this is actually like a what made them, just the most savage warriors on the face of the earth is that like yeah, they've got the factory in house for the berserk sauce. But like like there's a I guess like the creativity and like the artistry that you're talking about, like I like to think that it would be, a gent like that there's got to be like a gentler sort of face of that coin, that there's maybe some bacterium out there that is through some other mechanism producing just the MAOI,
Hmm.
that is not producing the static, but that is just turning up the volume on the symphony. Because that is like to me like what screams like I guess striking the balance there. But maybe
Yeah.
maybe there is no maybe there is no balance to be had, and it actually is like the line between genius and madness is very thin and all of that. Like
I
who's to
suspect
say?
in practice you have a subset of your population that goes into these extreme divergent neurological states, and usually they're just crazy, but occasionally they try making a stone axe a different way and it works better, and then that travels back into the rest of the community. I think that might be the
Mhm.
secret for modern human behavior, that we have
Is
a
like the
way.
the edge cases
Yeah, yeah,
and
yeah. We have a
creating
way
like
of allowing those kind of weirdos to do unusual things and then pay attention if it actually works.
well, this is what dudes are for, right? Like this is what I actually think there's something here because it's like like well, I don't know, like the I guess it's not well, okay, I don't know. It's maybe not quite as much like that as I used to think because when the idea was like okay, the microbiome is like well, actually it is still largely mother to infant, right? And
Mm,
like to that extent, women are the keepers of this like eternal flame kind of thing. They've got to maintain a relatively stable microbiome. They've got to like be there and pass it on to the children, right? Dudes are allowed to like be a little weirder, right? We're allowed to, maybe not yeah, like you're allowed to introduce some new species that are maybe not core to the microbiome or whatever to have some variance that if it works yeah, maybe it's selected for, but if it's not, then like, well, you're out of the race. But
Mm,
hey,
mm.
not where that came from.
I mean,
So,
well,
uh,
that brings up a general question about like the trade-offs with microbiome stability and flexibility and the possibility that there are already pre-existing mechanisms where organisms, animals under stress kind of spin the wheel. They increase the variability of their microbiome to see if they can
mm.
find a solution to the problem they're facing. Do you have any thoughts about that? That there might be ancient mechanisms for like evolving our microbiomes?
Oh yeah. Well, I mean, just the desperation itself is the engine for that. I would say.
Mm,
Well, and so I've got a post on this, right? It's called, I think, Cheating at Evolution.
mm,
And it's about how like the microbiome is a sandbox. It is an evolutionary sandbox in that like if a mutation, in order for a fit mutation to end up in every cell of your body, right? Let's say, a liver enzyme mutation that like makes it more efficient at degrading terpenes so that now you can eat pine needles just fine. And like
mhm,
that's actually like a healthy source of fiber for you and stuff rather than being like, I don't know, just like the terpene levels is probably too high for most people. Like in order for that liver enzyme mutation to, actually improve your fitness, like that enzyme has to be or that mutation has to be a germline mutation, right? It has to happen like to the gamete or like in, one of the earliest cells in the zygote or whatever, like in order to actually like end up in all the liver cells and be a useful mutation. If that mutation doesn't actually like is it's not a gain of function, it's a loss of function, as like most mutations are, because
mm,
the system is pretty finely tuned. Well, you're fucked. Like maybe now you've lost the ability to digest, like, I don't know, if you break an important enzyme, the like one of the cytochrome P450s, you can end up exposed to a lot more environmental toxins and things like
mhm.
that, right? Um,
And you only
it
get so many like individual offspring per animal, so you get very few chances to get it right.
Yeah, yeah, exactly. And so, but now you think about a similar mutation happening in a gut bacterium, right? And if that mutation works, if it is a gain of function mutation, suddenly, like the only, like, I guess what I'm saying is that in each of your gut bacteria has like at least 4 million clones of it, right down in there with it.
Mm,
And if one of them has a mutation and it's a fit mutation, then the only difference between it and its 4 million closest relatives is that it is fit. Like it will come to dominate the microbiome. It'll outcompete them. And because the things that are, toxic to a bacterium tend to be the same as the things that are toxic to human, not always, but like generally speaking,
mm,
like if, yeah, like there's a way for natural selection to allow the evolution of a trait, a new trait, a fit trait within one host generation,
yeah.
because 2,000 bacterial generations can happen in a couple weeks, right?
And then
If
that
uh,
becomes reliably transmissible through the mechanisms that pass the microbiome on to your offspring.
Exactly, yeah.
Yeah.
Yeah, and but the flip side is like if it's a loss of function mutation, if that like mutating that enzyme breaks it, uh, this bacterium will die, and one of its 3,999,000 closest friends will take its place, right?
Mm,
And it's the way I put it in the post is that like natural selection is a progress engine that runs on death.
hmm
Right. And so the smart move for guys like us is to adopt some creatures that have a much shorter generation time and which, yeah, it's not such a big fucking deal if one of them dies.
mm,
And we can sort of outsource the dirty work of trying and testing this evolutionary, roulette to them.
mm,
Uh, yeah, so I don't know. I think there's a lot of power in that, in letting that do its work. And so if like you want to sort of put the evolutionary pressure, so to bring it back to your question about like times of desperation and spinning the wheels to accelerate that, it's like, yeah, when your crops fail and you find yourself eating a lot of fucking tree bark this winter,
Hmm.
you're going to be selecting for bacteria that have xylan degradation capacities, right? That can break down tree bark and turn it into useful nutrients. And if you don't have any of those genes in your gut microbiome, guess what? There's going to be some on the tree bark because the bacteria that live on the tree bark or whatever fungi, like they have spent the last 30 million years figuring out how to get useful nutrients out of it or like, degrade the toxins in it or whatever, or else they wouldn't be there. And so, because a lot of bacteria in the human gut are naturally competent and can take up genes from the environment just like wholesale, uh, that is sort of the recipe for it's like if you want to get better at eating tree bark, you eat a lot of tree bark and you're likely to sort of acquire that fitness. It's a little bit of Lamarckian evolution, right?
Hmm.
Of like, giraffes got long necks by stretching them a lot to reach the tree tops. Like he had a point,
Yeah, yeah.
in some ways. It wasn't quite right, but like there's actually the more we understand, the more we learn, the more we come to understand maybe he was onto something with that, which is pretty cool.
Well, I mean, this is my final question I want to draw your opinion on. That if you look at the history of life on Earth, that the mechanisms for evolution are themselves always evolving. And the example I always point to is the symbiosis between pollinating insects and flowering plants led to a massive explosion in diversity and function in both of those halves of the equation.
Mhm.
And I wonder if human beings have the potential to become the universal symbiont for accelerating the exchange of evolutionary information, of genetic information of across all species, to basically enhance that process, that connection between all organisms that happens through horizontal gene transfer, and on top of that, microbiome exchange.
Wow. That's like a vision for the future, man. That's a great vision for the future, like of, sort of like and I guess this kind of circles back to like one of your previous questions about like what can plant or animal breeders do, right, to sort of take advantage of that microbial and it is just like yeah, take the good looking healthy cow and feed its shit to the sickly looking one and like see if it doesn't like to the extent that we can shepherd and steward these internal ecosystems towards the state that we want to see, like, yeah, I mean, I think there's massive potential to like, I don't know, to take this natural path of evolution and become the instrument of it, right? To sort of, I mean, to bring it back to something from one of your previous episodes, to like the leafcutter ants, right? Like
Hmm.
to do that sort of same thing that the leafcutter ants are doing, but in a very, very deliberate and informed way, I think there's like absolutely huge y- I'm gonna like bring it way out of left field, but like, are you familiar with ambergris?
Oh yes, yeah, yeah. The resin that whales vomit up that gets turned into
Yeah.
perfume.
Yeah, what is that?
It's a cholesterol derivative, isn't it?
It's like I don't fully recall, but it is like a weird polycyclic something or other, and like I'm almost positive that it's a fermentation product.
Yeah, it's like
Like,
it's kind of like a kidney stone, but of some other metabolite. But
yeah, I will,
micro
and
that.
I think it's like squid beaks is a major part of it or something like that.
Mm,
But there's definitely like the possibility of like and this is something that has just been like at the back of my mind for like if I need funding and this poop stuff thing, like if this poop stuff doesn't work out, of like you could probably produce artificial ambergris if you can like figure out what microbes in the whale's gut are doing that thing. And that's, just sort of the beginning of it in terms of like that's the most profitable thing to do because that stuff retails for like $20,000 a gram or something, right? Like, because it's, you can't like go and kill whales to harvest it anymore. And so you just gotta luck out and find some washing up on the beach. But like somebody has, done the microbiome analysis of those, and there's some signals there. It actually it's not implausible that we could have that kind of fermentation technology. And I think there is like, I don't know, that's sort of the trivial case, but if
It's
we
the
can.
microbial's philosopher's stone. You're turning squid
Yeah,
beaks into gold.
well, and that's, I've joked with my friends of like because it's like and we were talking earlier about like the economics of making a single dose product viable, and it's like the inputs here for the astrobacillus at least are like basically bouillon and offal, like the worst part of the pig.
Mm,
Nobody wants the brains. It's like, $6 a pound at Klaus's German Sausage and Meats because yeah, who's eating that these days? But it's like you're taking that. It's like to take something that's like, $50 a liter at the maximum to produce and turn it into like a value add product where it's like actually we are, like maybe a dose is 100 milliliters, and it's retailing for $6,000 for a dose. Like
mm,
it's unbelievable. That's alchemy. That is
hmm
turning, shit into gold, and Rumpelstiltskin could never, like it's but because what we have potentially here is, it's something that is spinning these things into that most precious of commodities, which is time,
hmm
right? Human life, like
Yeah.
quality and quantity of years, right? Because if we can prevent a heart attack or a stroke, like that's gold, dude. Like
Mm,
not even in the literal sense, but in the metaphor, like what else are we here on Earth for if not to like make life better and easier and longer for one another and more time with loved ones and shit like that. So
mm, mm,
I don't know.
hmm
I'm very optimistic about like if we can get these technologies unlock and actually like then I think it will be an epoch defining thing in that it'll be like pre I hesitate to say like a transhumanist kind of thing because I think it's sin humanism. It is like adopting it and embracing the malleability and the aspects of our humanity that are non chromosomal,
Yeah. Yeah.
but like it will be, I hope, yeah, like really a before and after kind of thing where we can get most, if not all, chronic diseases like on lock the way that we got rid of measles, you
Yeah.
know?
And,
So,
and it's a worldview that grounds your humanity and connects it to the rest of the world, I think, in a way that's quite beneficial.
yeah. Well, that's two more thoughts and I don't know, then maybe I'll stop yammering, but like I think there's one bit from a piece that I have written about oxytocin and the microbiome and the role there, and it's like, you know, a part of what oxytocin does in the body, and because it looks like there are bacteria that can sort of hit that button. This was the work that I did at Erdman's lab at MIT. Uh, uh, like, there's a bit at the end of the piece about like how oxytocin is not so much enabling altruism. Like, it's not so much enabling altruism and selflessness as it is letting us expand our definition of the self,
Mm,
right? To include others like from our tribe or whatever, and to see, yeah, to sort of see others as part of that and take maybe the selfish action, but it's a broader self. And that, uh I guess it's yeah, like a worldview that I think is a little humbling and sounds suspiciously like enlightenment, right? To view yourself not as like an individual, but just like as a sum of parts and part of a greater whole,
yeah.
you know?
Yep. Yep.
Um,
Absolutely. I
and
love it.
yeah, well, we can leave that. I don't remember what the other thought was going to be, but
That's okay. Maybe I'll have to get you back in a few years' time when you've pushed things along a bit further. But to wrap off, do you just want to let people know where they can follow up
oh,
and read
yeah.
more about your work?
Yeah, again, the blog is Eat Shit and Prosper. I don't think anybody else on the internet is using that phrase yet. So you can just Google that, you'll find me. But it's Steven Skolnick at Substack or .substack.com if you want the more direct link. I'm on Twitter, but I'm giving up on Twitter, so don't just like sign up for the mailing list. You'll get the emails directly to your inbox. If you're in the US and you have had your blood work done in the last year, Sign up for the cholesterol coprostanol study. I'll mail you a poop sampling wipe, and you can, see if you would be a good candidate for the cholesterol bacterium because we are about to get that off the ground.
Excellent.
And if you or a loved one has like schizophrenia or something like that, email me. We'll get your microbiome sequenced, and we'll see if there's anything we can do. I
Mm,
think that's the takeaway here is that like this stuff is a lot more in our control than we I think previously even suspected, and that's a scary thing to grapple with. I think a lot of people just assume that it can't be true because the gravity of our inaction, the implications are like there's a lot of responsibility there, right? But
yeah. Older
if we can't
generations
think
have
about,
definitely been conditioned to just wait for a guy in a white coat to tell you what to do. And I think
yeah,
younger generations are starting to leave that attitude behind.
yeah, yeah. Well, it turns out you can just buy white coats. So Hey, yeah, Shane, it's been great talking to you.
Yeah, I'm
And
so glad I got the opportunity to share you and your work with a wider audience because I think everyone can benefit to get a bit of a view about the kind of stuff that you and people in your field are working on. Thanks for listening. For another interview on the power of microbiology, check out my conversation with Bactelium. Link is in the show notes. You can subscribe for free for my weekly posts on Substack, or grab a paid account to support my experimental farming work and unlock the archive of past posts. Also, check out my book, Taming the Apocalypse, which explores the potential for low-tech biological technology to build a post-industrial civilization. In next week's episode, I talk to Mark Christensen from the Heritage Food Crops Research Trust in New Zealand about his extraordinary work breeding nutrient-dense crops to boost his community's health.